Hi, Philip. the first speaker is having a little problem, so we may have to wait until the second speaker to get started. Is Mohammed, are you here? Is Ushi Patrick here yet? No. Abdul? Abdul Okansi? Amadou Wege is here. Is Mohammed here? Mohammed Nuruddin Abdul Karim.
Hi, Lawrence.
Yes.
We are still waiting for people.
Yeah, we're still waiting for people and a
Yeah.
couple of speakers.
Yeah.
Hello.
That means
Hello.
we have to start a bit late. Yeah.
Yeah.
Good
Okay.
morning, Philip.
Yeah.
Hi.
Morning, welcome.
Hello. How are you?
Okay.
Bet I am here. Do you want me to start?
No, what I try to do is keep everything on time. But I don't know. What time is it now? So
Okay.
none of the speakers before
No,
you are here.
my problem is that I hope I will start at 10 o'clock as it is written, because after I have to go to another meeting at the university.
Understood. I'm going to see if I can reach some of the other speakers.
No, Lawrence, I mean that at 9 o'clock.
What time is it where you are?
9 o'clock Senegalese time. I have to go to talk with African strategy for
Okay.
fundamental physics and application.
What?
And this is another Zoom, you know.
Right. What time is it?
So
Where you?
it is important for me to start on time here.
Okay.
The
What
meeting
time?
with ASAP is at 11 Trieste time, which is 9 Senegalese time. Now, presently, it is 7:16 here in Senegal.
Did you?
It is 7:16.
Trieste is GMT +1. You should check that.
Yes, I know that. GMT +1, which means 9 o'clock in Senegal. And South Africa is a GMT +2. So I am supposed to talk at 10 o'clock, which is 8 o'clock here in Senegal.
Okay.
Okay, so it's all right.
Hi, Ed. How are you? So let's see. We are still missing a speaker. Is Maxwell here? No.
Maybe we should use the available speakers to talk.
Okay. Professor Wege, are you available to start? That throws the timing way off, but well, whatever. He may have moved to
You want me to start?
yes.
Okay, so
Um,
um,
so you can share your screen.
yes. Let me make the check. Just 1 minute. Do you see my screen? Do you see my screen? Hello?
No,
I don't see your screen.
But I put it.
Let me see. I have a lot of windows open. Let me see what's
What
going
should I
on.
do?
Try again.
Yes, it's here. It's there. Oh, okay.
You are sharing now? I don't see you as sharing.
Yes, I'm sharing.
Can anyone else see his screen? I don't see his screen.
me either.
Do you see it?
I do not.
So I don't understand because here I see in my screen here they say partager. I put on partager and they ask me to open the parameter of the system. And what I have to
Maybe start over the process.
do. Reconnaissance vocale, accessibilité. Microsoft. I have a Microsoft PowerPoint. Do you see it?
I no.
I don't see it. I don't know if anybody else sees it.
But I didn't upload on the system.
Do you see a little green button at the bottom that says share? You have to use that.
I have to use what? Yes, I see the green things where it is.
And you should have the ability to share. Or you have the ability to share.
Tous les participants. Okay. Yes. I do it. Is it okay?
I don't see your screen. Let's start at the beginning. Let's use the green button at the bottom.
Yes, I click on the green button.
And how many computer screens do you have?
And when I click on it, I see just I see fenêtre application, Windows. I put partager, autoriser, yes. And they asked me to authorize the host, and okay, the host.
You may also share your presentation with the host so he can share on his screen.
Yeah, why don't you just email your presentation to me?
What?
If you just email your presentation to me, I will advance it for you.
Okay. But it is a very big file.
Try it. Try to send it.
Okay.
Have you sent it?
No, not yet, because I have to. No.
Maybe you have to reboot your computer to make the sharing work.
So I don't know, because I am on my web page. Okay, so let me try to go to the email and to send it to you. But I have seen you sent to me the talk of Omolulu, not my talk.
Right, that's not what we need, right? That's
Yeah, this is for Omolulu. So I send it.
Okay.
You got it?
I have not, hasn't come in yet. You,
It's a big file.
let me see if it went to spam. You sent it to my L Norris address?
Huh?
You sent it to lnorris.org?
Yes.
It hasn't come in yet.
Your habitual.
Is there a way that you could like compress it and then send it? Here, let me send you a Dropbox link.
Any other speaker around?
I don't understand why it's not coming. But even I am in front of my screen. My screen.
And you can't share your screen at all. Is that what's happening?
Yes, when I click on the green button, so I go to the page where it is written second camera, audio from the computer, fichier video. What I say, I don't understand. iPhone, iPad. I see here video file, second camera. I don't know what it means.
Well, now you're sharing your screen, at least. Or you're sharing your camera.
When I put second camera, it show myself.
Okay. Now you just need to share your screen. Are you using a PC, Microsoft, or Macintosh?
I am using Macintosh.
A tablet or a laptop?
Macintosh, a small one,
Okay.
laptop.
And so inside the Zoom app, do you see a green button that says share?
Yes, yes, on the Zoom, yes, I see that, share.
And then when you click on it, what comes up? This should be say share camera or share screen.
I go to share
Mm-hmm.
and I have full screen, but it's right desktop one with a alert signal. So I don't understand why. Okay, now I put Microsoft PowerPoint, so I will see. It's written Microsoft PowerPoint unknown.
Okay.
In my screen, I can't see myself.
Right. Maybe you need to turn your camera off and then see what happens with that. Let me see if your presentation. So here's your I get your presentation now.
Huh?
Right, it is a big file.
You can see it now.
It's still downloading. But it's coming.
It's coming.
Yeah. All right, I have it. Okay, so I have it, and I'm going to. So you need to stop sharing. And I am going to share. Yeah.
Okay. I can see it.
Okay.
Could you make it bigger?
Okay.
Okay.
You good?
Yes, this is fine. But you are going to give the slide.
Yeah, just tell me when to advance the slide. Can everyone see the slide now? See the presentation? I'll take that as a
It's
yes.
visible now. It's visible.
Yes.
Okay. Okay, you can go ahead and start.
Okay. Thank you very much. Good morning. Good morning, everyone. I am Amadou from Senegal, president of the African Physical Society. So I am going to present some application on laser spectroscopy, laser breakdown spectroscopy, laser LED, and X-ray induced fluorescence spectroscopy. Okay, next slide. Lawrence? No, I will start with a brief presentation of the African Physical Society. Next slide. As it is this joint conference of African Physical Society and African Light Source, so I just make a small presentation because at the meeting of ASAP, there will be a presentation of the African Physical Society. So I have just to say something for people who didn't know that African Society was launched its official ceremony in 2010 in Senegal, when it is incorporated in Ghana. And the main goal are to promote research, education, and training in physical science and application, to enhance technological, economic, and socio-cultural development in Africa, to promote effective contact and cooperation among African physicists, and collaborate with other physical society and international organizations in promoting scientific activity in Africa. And the African Physical Society has to serve as an important resource to all African physicists and help leverage their communication and collaboration. This is why we are always collaborating with, for example, with African Light Source in a many common joint meeting. Okay. Next slide. Next slide. Okay, this is just a photo when the delegate of African Physical Society went to the presidential palace, and the former president of Senegal is an honorary member of African Physical Society. Next slide. Next slide. Because I cannot run. Okay, go ahead. I just show very quickly the photo. Go, Lawrence, go ahead. Go ahead. This is the common meeting of African Physical Society with Ghana government for the International Year of Light in Accra in 2010, with the Minister of Science, Professor Alote, who was the first president of the African Physical Society, and many others. Next slide. This is the meeting in Kigali of the African Physical Society. Next slide. And this is a meeting in Trieste with at the IUPAP meeting. And here we are with the director of ICTP and the Nobel Prize of Physics, Philip Williams, and responsible of the Crystallography Institute in Benin. Next slide. And this is the last year, the meeting in Johannesburg, where they are giving the stick to Simon. The stick is coming from Ghana. This is a symbol of African culture. Okay, next slide. And this is also the meeting in Johannesburg last year. Next slide. Okay, now, as you know, this year is the International Year of Quantum Science and Technology. And as I'm going to speak about laser, you know, laser is one of the major components of the quantum science and quantum technology. Next slide. Next. So I say a powerful instrument in quantum technology and application. Next slide. Principle of laser, or maybe Lawrence, is it possible to talk from my PowerPoint so that I can change it?
What do you want me to do?
No, you will continue to do that, and I will talk from my PowerPoint.
That's fine.
Is this possible?
Yes.
Okay, let me do that. Okay, so I was saying that laser is powerful instrument in quantum technology and application. And just a few words about the principle of laser. It is the acronym of English word light amplification by stimulated emission of radiation. And the operation of laser is based on the theory of the emission and absorption of light by matter, which is formulated in 1917 by Albert Einstein, who got the Nobel Prize of Physics in 1920 for his work on photoelectric effect. And there is three main process for laser functioning. It is there is spontaneous emission, absorption, and simulated emission. Simulated emission is the main one. As you can see in the screen, this is the spontaneous emission and absorption. Lawrence, are you following?
Yes.
Okay. And here, this is the simulated emission. You know, from one photon, you can get two identical photons. And from n photon, you can get two n identical photons. But the problem is that the amplification of light is based on the simulated problem. But to obtain a laser in general, in addition to the stimulated emission process, it is necessary to create what is called population inversion. That is to say, to answer that the population of N2 of the higher level, if it is a two-level system, is higher than the population of the lower level, which is not a normal situation in a normal atomic state. So if you can do that, you can have a kind of avalanche of photon, and if you put all these medium in between two mirror, which one is 100%, the other is less than 100%. In a very small short of time, you will get a very strong radiation, which is a laser radiation. And the laser was I can say was invented. The theory of the laser was invented by two well-known scientists, Charles Towns, Alexander Prokhorov, Nikolai Basov. Alexander and Basov are from the ex-Soviet Union, and Towns is from USA. And I got and this is the first laser, ruby laser, made by Mainman, American engineer, in 1960. This is a photo from the New York Times, and it was a ruby laser. And I got the privilege to have a photo with Towns in China in 2005. Now there is many kind of lasers, and now we can go up to attosecond lasers. And last time there was a talk by Anne Willier about attosecond generation. Now I'm going to talk in my outline about the laser-induced breakdown spectroscopy, laser-LED induced fluorescence spectroscopy, X-ray fluorescence spectroscopy, and multispectral LED microscopy. So this is the photo of my laboratory at the university with a different topic. I have to say that we have also theoretical research in a resonance state of atom, in auto-ionizing state, and so on. So laser-induced breakdown spectroscopy. The LIPS is a technique. It is a fast and essential non-destructive diagnostic method based on the analysis of the spectral emission of the plasma produced by focusing an intense laser beam on the sample to be investigated. And LIPS technique doesn't require difficult sample preparation and can be applied on sample of arbitrary shape in solid, liquid, and gaseous state. This is the principle of LIPS. You have absorption by absorption of photon by electron, which is less than 1 femtosecond. You have a terminalization of electron, which lasts about 100 femtoseconds. We have the cooling of electron and energy transfer to atom and ion, 1 picosecond. Thermal diffusion again for 10 picosecond. Fusion of the material and ablation, 100 picosecond. Beginning of the plasma formation and ejection of heavy particle, 100 nanoseconds. And in the hot plasma, the ejected material is dissociated into excited ionic and atomic species. And this is what you can register, and you can see the composition of the material. This is the LIP experimental setup that we have used. We use a 10 nanosecond wide laser pulse is needed. It is a 200 millijoule Q-switch neodymium YAG laser. And it is designed to perform various functions safely and clear view of the sample. The chamber is an eyewear safe enclosure with safety interlock. And now what we have done, what application we have done with this laser-induced fluorescence breakdown spectroscopy, we make a measurement of pollution in the urban zone. One of the application is application in soil pollution. In Dakar, there is a bay of Anne where there is a strong pollution by industry, and we have made measurements there, which show very, very clearly that there is a pollution from mercury. And you know, mercury is a heavy metal, and it is very dangerous for the health of population. And of course, we show this result to the environmental ministry, and we tell them to take necessary action to supprime this pollution. And this is another thing that we made a measurement in the street of Dakar at the Azadirikta Indica in Dakar. The normal name is Nim, and we have seen then that there is zirconium. Even we have seen some zinc also in this kind of in the trees which are absorbing from the circulation and also from the soil. In Senegal we have a big production of zirconium even in some places at industrial level. But it is a heavy metal also, and it is dangerous. And this is another application. It is application in medicinal plants. In Africa, many people are the health system for many people is medicinal plant because they have no money to go to the doctor. So it is important to see from the indigenous plant what is the composition and here we were interested by Ocinium basilium, which is basilic, Moringa oleifera, Cambretum micratum, Kenkeliba. Kenkeliba is a kind of plant. It is for the breakfast. Every Senegalese is drinking kenkeliba. Simbo, Gotom, citratus. It is a Latin name, and we got the collaboration with the Department of Vegetal Biology. Citronelle, mint, menta, eugenia, and as you see, for example, we try to find the association of potassium, especially because potassium is an important component for diabetic people. You need to have enough potassium in your blood in order to absorb the glucose. And also we make also some application in dental analysis. And the objective of this study was to assess the spectral variation depending on the pathology of different patients affecting the dental mineral tissue, and also to establish a correlation between different lesion and the chemical basic composition of the tooth. Here you have the fingerprint of enamel. This is just by a shot of a laser on the enamel. You can get all these things. Even we got a signal from the instrument also, which is making the measurement. And here we have the spectral density in dentin tissue for nitrogen, for calcium, for carbon, for potassium, depending on the different on the different pathology. And now I will go to the laser-induced fluorescence spectroscopy. The LIFT is it is a widely used technique in research for a variety of analytical application, from interrogation of plasma plume to determination of cancerous tissue to fluorescence spectroscopy of single molecule. And it is a very sensitive approach available for analytical purposes. It is easy to implement, well investigated, largely non-invasive, and imperative for many applications in environmental monitoring. Here, this is the principle of fluorescence spectroscopy. When you excite an atomic system, which go to the higher excited state, there is a kind of a relaxation system. And after this relaxation to the triple one and triple two state, there is a fluorescence emission. And this fluorescence emission, you can get a lot of information. For example, for monitoring plant by spectroscopic detection of this fluorescence, it is a non-contact and non-destructive method. And the laser-induced fluorescence spectroscopy is an important aspect of active remote sensing. And as you know, for the development of plant is based on the photosynthesis, which when the sunlight go to the chlorophyll. And to measure the efficiency of this process, we have to measure the fluorescence. And the measurement of the depression of photosynthetic rate is important criterion in the evolution of accessory pigment to the chlorophyll A and stress condition in plant. And the absorption spectrum of chlorophyll is in the red and in the blue. This is why the plant are green. And here there is the instrumentation we have used for that. It is a small box with a blue diode lasers with a spectrometer inside, and with that you can make a lot of measurement in plant monitoring, for medical application, and for many other things. And there is a fiber optic. It is a fiber optic spectrometer which communicate with the PC via an external analog digital converter. Here, this is the application we have done with this system. As I say, the monitoring of chlorophyll, and also we make the analysis of many plants in Senegal. You can see some of the name here in the slide. And this is the schematic view of the experimental setup. There is an ocean optic the fiber optic, and with the fiber optic, you go to the sample, and you get back the fluorescence with this dichroid mirror. And another thing that we have done concerning fluorescence spectroscopy, it is the use of X-ray, a portative X-ray instrumentation. When you have monochromatic radiation irradiating atom of an element with subsequent electronic transition from a higher shell may be observed accompanied by the emission of X-ray photon. And the energy of the emitted photon is characteristic for the atom of the element, while their number is directly proportional to the concentration of the element in a sample. And we use this technique to see, of course, the composition, and also to see the concentration of mineral in Senegal, like concentration of gold, concentration of some of iron in many places in Senegal, in the Senegal Oriental. This is the view of the laboratory where we make this measurement at the Institute of Nuclear Technology. I was in the past the director of this institute, and I bring all this instrumentation to the institute. And this is an application in geology of X-ray fluorescence spectroscopy. You can make a characterization of rock sample regarding the petrogenic aspects. And here we have application in agriculture. We make some analysis of Artemisia. You know, there was a time where people are thinking that Artemisia is very strong for malaria. That if people say you drink malaria, you can. So we make some measurement for that also, and we make a machine learning and instrumentation, and to make very fine measurement. And here, this is also in other agricultural product. This is the spectrum of tea from Cameroon, tea from China. So you can make a qualitative analysis of different agricultural products. Now, concerning multispectral imaging, so it is spectral imaging. It is a combination of imaging and spectroscopy, where a complete spectrum is collected at very location of an image plane. Our experimental setup consists of a modified digital microscope, which have been performed by the Swanberg group at Lund. Lund, the same institute like Anne Lully. Even I have visited this institute many times. And what we have done is that these microscope have been modified in our different group in Senegal, in Ghana, in Kenya, in other places in Africa. And the microscope is a constitute of 9 light emitting diodes, corresponding to 13 different wavelengths. And you use the LED. The LED is not expensive. You can buy a LED for, I don't know, for $1, even less. And there is an illumination mode, transmission, reflection, and scattering. And there is a very broad band ranging from 375 nanometer to 940 nanometer, so that we can provide illumination in this different geometry. So the measurement with the multispectral imaging microscope, we make a characterization of Plasmodium falciparum, infected red blood cell for malaria monitoring. And for this purpose, we use a multispectral microscope to study optical property of red blood cell in diffusion and transmission mode. In addition, we use machine learning technique for the differentiation of healthy and contaminated red blood cell. And the main aim is to get a quick malaria diagnostic tool. As this is the microscope that we have in our laboratory. And there is a computer interface with a LabVIEW medium, and we use MATLAB to make all the spectral analysis. And this is the component of the multispectral microscope. You can see here all the different wavelengths that we have used. There is many wavelengths, and it is affordable for many laboratories in Africa. This is my student working on the system, and here this is the result in transmission mode and the result in scattering mode. And of course, we have noticed that in scattering mode, the results are better. And here, this is the optical spectra of infected red blood cell and healthy red blood cell. Yeah, you can see the difference in the different mode, transmission mode and scattering mode. And we make the measurement near the ultraviolet, visible, and near infrared. And this is the support vector machine learning. This was extremely important because it is important to make a differentiation between infected cell and non-infected cell. And for that, we have a function, we have a cost of functions that we have here that we can see. And we have a sample with known parameters, and after we make a test with our blood sample. Here you can see the sensitivity, the transmission in the transmission spectra. You can see the cell with plasmodium in the red, and without plasmodium, which means the healthy, the infected cell in red and the non-infected cell in blue. And you can see that in this. And we obtain this thanks to the machine learning utilization. And here, this is also machine learning for differentiation of healthy and contaminated. This is in diffusion mode. And here the sensitivity is about 96%, with a specificity of 83%. And here you can see the contaminated cell in the top, red one, and the non-contaminated in blue. So thank you for your time, and many thanks to our partner and sponsors. It is ICTP, AfLS, AfPS, ALS, ISV, it is the International Science Program in Sweden, the LAM Network, the government of Ghana, the government of Senegal, and of course the University Cheikh Anta Diop of Dakar. Thank you very much. So Lawrence.
Okay, was there, let's thank the speaker. And then let's see if there are any questions for the speaker. So if you have a question for the speaker, you can just unmute yourself and ask. Or you can put it, let's see, there's some questions in the chat, comments in the chat. Any questions for the speaker? I can ask one question if I can.
Yes, of course.
So the example you showed of medicinal plants, were you able to isolate the compound from the plant that was medicinal in nature? So you're able to.
We don't go to this stage, but what we have done is to show what is the composition of plant and what is the active element, which we can show to the to indigenous doctors that this is the active element, the active which can be used for diabetes, for other diseases, you know. And this is important, especially when we make analysis of Moringa oleifera. You know, here in Senegal, many people, they think that it is good for diabetes, but they don't know why. And we make a measurement and we show that there is a lot of potassium in the Moringa oleifera, and the potassium is one of the elements which help for the absorption of sugar in the blood, in the cell.
I see. Well, how I mean, so I guess you have prior knowledge that the potassium was the important for the absorption. You know that from pharmacology, and you just have to show that there's potassium in the sample in the plant that you're analyzing. Though it could be something else entirely, right?
Yes, of course. What you say, it is important to go ahead, to extract this and to measure the concentration also. You know, in our lip system, it was possible to make a measurement of concentration, and we have some result on that, but I didn't show it.
Okay. Okay. Well, thank you. Are there any other questions for the speaker? Okay, Professor Wege, we know that you have to move to another event.
Okay.
And what we can do right now is we're gonna flip the order of talks. Ushi, are you ready to give your talk? Ushi Patrick?
Yes, I'm ready. Yes, I'm ready.
Okay. So you can start your talk right now.
Okay, let me share my screen. I'm sharing my screen.
You are sharing your screen?
Let me go there. Oh. Not coming up. Can you see my screen? I don't know.
I cannot.
Okay. Can you now? No.
No.
Serious.
Is there something not right with the settings?
What is the problem?
You should have the
Okay.
sharing.
Yeah, more
Oh,
than.
here you go. Yep. Perfect. Okay.
Okay.
You can start.
Okay. Thank you for this opportunity. My name is Shea Patrick from Nigeria, and I work in Cross River University of Technology, now University of Cross River State. And my title is to minimize this background is covering my screen. Okay. Assessment of background ionizing radiation and selected dump sites in Calabar, Cross River State. And as I earlier said, in Nigeria, I work with the Department of Physics, Faculty of Physical Sciences, University of Cross River State. On my outline, I have introduction, I look at other related works, look at the material, the results, conclusion of my findings, and the materials I contacted. In Calabar, there's rapid urbanization around the community, and this has caused several pollution around the environment. And ionizing radiation, one of those radiations that cause serious havoc within the community, within the environment. Higher population of medication in hospitals now is either it's cancer related or from pollution. So I took my time to look at pollution around the environment, especially dump sites. I look at other work that have been done in that area, though not in Calabar, and see their output so that I can also relate it to mine to see if what I've done is really in line. Olufade did that work in 2021, and his result was indicate that elevated radiation level in dump site environment with significant variation observed based on waste composition and management practice. Zilt did some work in 2020. Abdurrahman and Mohammed did some work in that area too. The materials used for this work is the chamber radiation survey meter model 451P ion, a GPS so that I can actually navigate the position or plot the position in a general map. I also use a handheld digger to excavate the soil for sample test and sterilized container, and those samples were tested using atomic absorption spectrophotometer. Yes, our results from what we took to the lab and the measurements we did on the field, within the first dump site in Calabar, we took 15 different samples. 15 different samples in 15 different sample points. And on the table, you can see our results in microsievert per hour. We plotted this to see what is the variation within the different sample points. And we see that the background radiation in those sample points, we have a peak sample point at E2 and as it changes. And the second dump site, we took sample points, about 13 sample points to get our readings in microsievert per hour. And that is the result we had within that dump site. The peak radiation is around G11, the sample point 11. So the table 1 and 2 represent the gamma radiation dose rates measured at each dump site location. The result indicates considerable variability in radiation level across the sample sites. It can be seen that dump site 2 possesses a higher level of ionizing radiation, its highest average being 3.5 microsieverts compared to dump site 1 being 2.6 microsieverts per hour, respectively. These findings correspond with the previous research work stated earlier, highlighting dump site as one of the major sources of environmental pollution or radiation contamination. According to the tables and figures above, showing the different radiation dose level, it is agreeable that there's an increase in the level of radium, thorium, potassium at dump site 1 and dump site 2. That is indicative of potential health risks associated with prolonged exposure to ionizing radiation. Such exposure can increase the risk of adverse health effects, including cancer, genetic mutation, and other radiation-related illnesses among population residing near those workstations or those dump sites. Although this effect might not show immediately, but this damage is certain after a very long period of time. The variability in radiation level among these dump sites underscores the importance of tailored remediation and strategies and ongoing monitoring efforts to mitigate environmental and public health risks associated with radiation exposure. The second dump site is close to a medical facility, and that is where it shows so much increase in radiation within that dump site. So meaning that medical facilities too can pose a radiation health effect if not well managed. So effective management practice, including waste segregation, contaminant measures, and periodic monitoring are essential to reduce radiation hazard and protect both ecosystem and human population in affected areas. Based on the measured gamma dose rates and radionuclide concentration, an assessment of radiation safety was conducted following international guidelines and regulatory limits. And that is how we got that result. The result indicate that inhabitants living near dump site 1 and 2 may be exposed to certain level of ionizing radiation, warranting further investigation and potential remediation measures to mitigate this health risk. The data analysis revealed several key findings. The dump site 2 consistently show a higher level of background ionizing radiation compared to dump site 1. This observation suggests that historical waste disposal practice and the types of waste disposal may have influenced the extent of radiation contamination at each site. Just like I said, dump site 2 is around the medical facility. So it had to be controlled. The dump site had to be controlled for radiation exposure. The gamma dose rate measurement collaborates the soil analysis findings because this is soil analysis from those dump sites, highlighting dump site 2 as a potential flashpoint for radiation exposure due to elevated radionuclide concentration in the soil. The variability observed among sites underscores the complex nature of radiation contamination in those dump sites or the environment, influenced by factors such as waste type, soil properties, and site management practice. So in conclusion, the study assessed the level of background radiation in 2 major dump sites in Calabar South area of Cross River State. The level of background radiation for dump site 1 ranges from 1.92 microsievert per hour, which is the lowest point, and 2.6 microsievert at the highest point, while dump site 2 recorded from 2.07 microsievert per hour as its lowest point, and the highest point 3.5. By this, it can be understood that the background radiation for each of these dump sites is slightly higher than the approved radiation exposure of 1 microsievert per hour for public health, set aside by the International Commission on Radiological Protection. The research finding underscores significant environmental and public health concerns associated with background radiation in dump site environment within our study area, which is Calabar. Elevated level of background ionizing radiation, particularly at dump site 2, pose potential risk to both the environment and nearby communities if proper safety measures are not taken and adhered to. By addressing these findings through targeted interventions and informed decision-making, stakeholders can work towards safeguarding public health and promote sustainable development practice within and outside our place of research or our research points. So these are materials we consulted for this work. Thank you. As I expect your input. Thank you.
Thank you. Let's give the speaker a round of applause for his presentation. And if you have any questions for the speaker, you can unmute yourself and ask. I have just one quick question. So in your work, did you ever become involved in any type of enforcement action for medical waste dumping?
No, I've never been. I've never been part of enforcement player in waste dumping. But I think these results can be forwarded to the Ministry of Environment for proper care of those dump sites. Most especially, they should not be close to a living environment as they are now because of what we have discovered from them.
And the isotopes that you found, what's their half-lives? I might have you might have shown a slide on that. I might have missed it.
Well, there's a further investigation that we are planning to do on that work again.
Hmm.
Because, at this point, we didn't go to find out what's half-life of those isotopes yet. But we're thinking that it should be a further work on that research. What we just did was the background radiation. We just discovered that the background radiation is elevated above normal for that particular environment. So
Right.
further dig to see if we can get discover the mineral or the ion contents or the radiation exposure from what materials in particular. Because we discovered that dump site 2 is close to medical facility, meaning that there are some particular isotope they are dumping there.
Yeah.
So our further research is to dig into those isotopes now and bring them out and see what kind of material they actually dump in that place.
And how far are people from the dump site? Are there like homes
Less,
next to it?
yes, less than 500 meters.
Oh, okay.
Less than 500 meters.
Okay.
Mm.
Any other questions for the speaker?
Yeah, I want to ask something small about it.
Okay.
Okay.
So thank you. Thanks, presenter. I'm asking one. What was the criteria of selection of the dump sites? Is it random sites, or are there specific interests in your sites that you selected? What's the information behind that?
Yes, one is random. One is random. Two is because of the nearness of that particular dump site to a medical facility.
Okay. Okay.
Yes.
Thank you.
So,
It'd be interesting to test the people that live around the dump sites.
for now, we have not.
No.
It's research in progress. We
Right.
have not for now. We are thinking of going to do a survey,
Right.
and in-source survey, speak with the people and hear from them.
Okay.
Yes.
Well, let's thank the speaker again.
Thank you.
And I don't know if there were some speakers that we did not see earlier. Is Maxwell Quartang or Muhammad Abdul Karim? Okay. Well, so we're scheduled actually to be on break, but let's see if the other speakers are here at the moment. Is there a Tessafe, Harisha, Neha, Lalorata, Lalostra, and Gideon Benton? They all have a few minutes to get here because the next session uh, will begin in 15 minutes.
Lawrence?
Yes.
Yeah, I'm thinking maybe we should send a reminder to the speakers for the next session.
Yep, I did that. But let
Okay.
me do that. You'd be surprised how many reminders some people need.
All right. Yeah, that'll be fine. That'll be fine.
Okay. So let's reconvene in 15 minutes.
All right then. It's okay. Thanks.
Okay, I want to make sure that the speakers are present. If you're a speaker in this session, please let me know. Is there Testafe Horisa here? Is there Nahalat? So Testafe Horisa, are you here? Okay, looks like we're missing a couple of the speakers. And the last speaker, Gideon Bentham, is not able to move his talk forward. So what we have here is dead time. Philip, are you still here?
Hello? Hello?
Yeah, hold on.
Hello?
Yeah, Dr. Norris?
Yes.
Yeah,
So
please,
you can hold on, you can share your screen.
But I think there's a lot of waste.
Try to share your screen and you can start your talk.
Which causes a lot of pollution.
okay.
For me, at least the medical waste is just they are concentrated at some points. You have some hotspots, although they are more dangerous. But you have the domestic waste, which are not just waste.
Please, can you see my screen?
Like
Yes.
innovate on how
So you need to go into presentation mode.
Okay.
Yeah. Thank you.
Thank you. Thank you for the idea. I think this is something also we thought about. So we're thinking that at some stage later, also community, local government authority can.
You put that in presentation mode
they can
down at the bottom where the little screen is.
Accelerators for the general.
I'm coming. My laptop is running slow. I'm coming now.
Perfect.
Is it okay, Dr. Norris?
Yep, go ahead.
So, good morning, colleagues. Please, my name is Gideon Bentum. I study at University of Johannesburg, and today I'm going to present on comparative analysis of camada rock using activation technique. And this work is collaborative work by Hendo, Nikamaza, and Tim Brooks, Samuel Cornell, Martin Koch, and Joe Hammond. So this presentation, we are going to discuss on the introduction, description of Mempe technique, lesson diamond pits and Kimberley selection, description of the AfLS 100 electron beam, activation analysis, high purity germanium detectors and data acquisition, detector calibration, peak analysis, and then we are going to present the results, which is residual activity, and we'll do the discussion and conclusion as well as the references. So when you talk about activation analysis, it's the process of using energy beam. The beam can be electron, the beam can be electron, photon, ion, or neutron source. And this beam is used to irradiate sample, and further analysis will be done on it. So with these techniques have been used in manual rich sample, leading to the right selection of techniques for extraction. So camada rock are the primary source of diamond, and detecting such diamonds and the background radioisotope requires special analysis. This rock exhibits spectral lines of long-lived radioisotopes when gamma spectroscopy analysis is performed on them. When they are not irradiated, and selecting the kimberlite with the diamond content in it becomes impossible. And I'm going to explain that during the experiment. So when similar rock is irradiated, it produces spectral lines of radionuclide that fall within short life, medium life, and long-lived radioisotopes, selecting the right kimberlite when diamond content within a time frame is possible. In this work, unradiated kimberlite and radiated kimberlite will be compared using activation technique. So let me briefly describe the Memper technique. In the Memper technique, a kimberlite rock of size 10 centimeter top is crushed and then irradiated with high energy beam photons to transmute stable element into PET isotope. What it means is that carbon-12 is going to receive the gamma beam to knock off one of the neutron and then to produce unstable carbon-11, which can be detected using detectors. So after this process, then we hold it for 30 minutes so that some of the radiation will reduce because its radiation decays as time elapses. And then after that, we are going to detect the 5K11 photons with the detectors. Why the 511 keV detectors? Because during MIMPET process, we want to construct image and the image will come from the 5 keV photons. So here, that's why we done the activation here. An activation in 40 MeV gamma beam. We hold it for 30 minutes and then we do, in this work, we are not going to discuss more about the classification and image reconstruction. And such approach can be used to detect diamond in the kimberlite at the same time gold. By now, I don't have much data for gold analysis. So the kimberlite was taken from Lesson diamond in Lesotho, and the diamond has an altitude of 3,100 to 3,200 meters above the sea. It is the one of the highest diamond mine in the world in terms of elevation. So if you check here, you see the diamond, Lesson diamond pits and main pet team. So if you see me there, I'm the one holding a phone and take it selfie there. That's where we got the kimberlite sample from. So Lesson pits. So this is the rock that we use for the experiment. So this experiment, we label the rock S11 and K6. The S11 kimberlite was irradiated and the K6 kimberlite was not irradiated. So with our previous analysis by kimberlite composition by SRF, that's the X-ray fluorescence, we got the concentration of the kimberlite to be 5%. And this one gives us what it was done for gray kimberlite, ultra black kimberlite, and we do the average to get the composition of the kimberlite. And the S1 rock is called mantle xenoloth. So this is the description of Ahu 100 MeV electron. So the electron has 1D and 2D, have asterisk D and D. The asterisk D is the booster, and the asterisk 2D is another ring that propels electron. But our experiment, we use only the electron injector, which is indicated here, electron injector parts. That's the only part that we used. And the asterisk D has a circumference, it has asterisk is 580 MeV with current of 20 mega amps, and the circumference is 45.7 meters and 40 meters beam. So this beam, the energy is high, then we have to create brainstorm photons from this beam. So here I'm going to play a video for you to see how the electron beam is operated at Aarhus. So if we check, this is the R-straight 2D, and this is how it operates. So if you check, that's the location of Aarhus laboratory at Denmark. And this is how the diagram of the facility. So the first portion here is the electron injector. So it will inject electron and go through the booster, which is the astrate. It will go there, and then after that, it will go run the booster, and then to eject it to another astrate 2D, and then it will eject it. So with this, we didn't use the ring, the astrate D and the astrate 2. We use only the electron injector part. So this is how it operates. And after going through the astrate, it will just prepare it and then it will eject it out. So that's how it works. And this one here, down here is the injector part, that's the electron injector part of the AfLS 100 MeV microtron injector. So the next one is the Kimberlite was irradiated with that with the beam, and then we do what we did was it was irradiated for 10 minutes, and the distance from the beam was 750 millimeters. And after irradiation for 10 minutes, then we cool it for 10 minutes, and then we detect it at 23.5 minutes. So if you take the K6 sample, because it was not irradiated, so there's no distance and there's no irradiation time and there's no cooling time, but it was detected for 1 minute, 49 seconds. So with the process of producing Bragg-Solon photons from the electron injector, we use 2.5 stainless steel and 5 millimeters copper plate flange to create Bragg-Solon distribution to irradiate the Kimberlite. So the diagram here, the first one is the beam source, is the electron injector coming from the accelerator, and here the Gen 4 simulation showing how when the beam passes through the targets and then producing Bragg-Solon photons. So the green is the photon, the blue, the green is photons, the green is the blue is the green is photon, and so these colors are charges. And you see that the red is negative, which is electron, the blue is positron, and the green is photon. So after detecting it with the high purity germanium detector, this is the sample. The S1 was placed in between 3 germanium detectors to get data for analysis, and the cases was also placed in a high purity germanium detectors to also get the data for analysis. And this is the CAN digital modular that is connected to the high purity germanium detector to get the data for analysis. And then after that, we use European source to calibrate the detectors to get a well-defined peaks. So this detector has a dead time of 0.4% and efficiency of 0.159%. So that is the dead time for the high purity germanium detector and its efficiency. So after doing the calibration with the S11 rock, we got a well-defined peaks, and here we don't know the exact radioisotope associated with the numbers in the S11 rock, and at the same time we don't know the number, the radioisotope that are also in the K6 sample, that's the K Kimberlite. And after using the European source, you got a well-defined peaks, and now we have to also identify all these peaks so that we can associate them with the exact radioisotopes that is showing that those particular peaks. And the main reason why we choose European 152 is that it has a longer half-life of 13.5 years, and It allows both energy and efficiency calibration with a single source, unlike cobalt 60 and other that need other isotope to achieve that. So this shows the spectrum of the calibration with the European source, European 152 source. So for us to identify the peaks in the spectrum, we use the nuclear database where we search the energy with specific half-life. If we search it, you are going to get the exact peak that is showing that kind of energy. So you put the energy and you put the energy and then you search. So when we search for energy of 1434, we notice that that particular peak is from magnesium 52 and V52, the 52. So we did all of them for all the peaks to identify all the elements that is showing those particular peaks in the spectrum. So our results, after doing the peak identification, we noticed that the first peak was 511 keV PET, and the 1369 keV is from magnesium 56 going to sodium 24. So we got the identified elements to be this radioisotope. So we say that 5 keV has a half-life of 20.3 minutes, and the remaining isotope are of radiological concern. It means that with MIMPES technique, our main interest is the 5K11, because that's where we can construct image to see indeed if there's diamond in the rock. So you say that short-lived isotopes, magnesium 56, magnesium 52, sodium 24, and cobalt 56, which are occupational hazard. Those radioisotopes are occupational hazard, and whenever you are in a nuclear facility, you have to be more concerned about it. And the long-lived isotope, which is potassium 40, is the environmental hazard. So that is for S11 rock. This is the element that we identify in it. So let's compare the cases, kimberlettes, and see if we are going to also get some radioisotope in the spectrum. So with the result of residual activity for the K1, we notice that 5 kV from the is coming from the thorium series. And if we check the peak is smaller as compared to the S11 peak. And what are the main reasons why this is arising? I'm going to discuss that. And we also saw 1461 keV, which is coming from potassium 40, and as it's appeared in the previous spectrum. And we have also hydrogen, which is 225 keV in the spectrum. And we also got thallium 208. The thallium 208 is showing because this is the background source at the lab during the experiment. So let's discuss the results. So if you take the S1 kimberlettes, you see that 5 keV PETs. We notice that 5 keV PET, magnesium 52, magnesium 56, sodium 24, cobalt 56, and potassium 40 with half-life of 20.3 minutes, 2.578 hours, 5.591 day, 77.24 days, and 1.248 times 10⁻⁹ years. These are the elements that we identify in the S1 rock. So we notice that 5 keV PETs, magnesium 52, and sodium 24 are short-lived radioisotopes. potassium 40 belong to the long-lived radioisotopes. And apart from KEV radioisotope, which is environmental hazard, the rest of the nuclei are occupational hazard. And then if we check the cases, kimberlite 5.1 KEV observed with shorter peak as compared with S11 rock. And we also notice that stable hydrogen element was also seen. And thallium and potassium were observed in the spectrum, and the thallium-2 was from the background source from the Aarhus lab. So what do you have to say about this analysis? Two Kimberlite samples, S1 and cases, have been compared. Potassium 40 was observed in both spectra with a half-life of 1.248 times 10⁻⁹ years, which is an environmental hazard, with hydrogen being the only stable element in both spectra. The height of the 5 KeV PET in the S11 rock was seen to be longer than that of the 5 KeV in the K6 rock. This observation shows that the 511 KeV, which is the main event for image reconstruction, can only occur in an activated sample at the time of 20.3 minutes, though it is present in unradiated kimberlite because of supernova processes during the rock formation and construction, and constructing an image from it will not be the desired result, will not give you the desired result. Also, radioisotope of half-life of minutes, hours, and day are of hazard concern. So it means that if you have two kimberlite samples, the one that has been irradiated and the one that has not been irradiated, if you want to construct image from those samples, the one that has not been irradiated, you are not going to see any meaningful diamond. You are not going to see the diamond in the rock. But with the activated one, since we activate the 511 KeV in it, we are going to use that one to get back-to-back photons to construct image from it. And these are the reference for this work. Thank you.
Okay, thank you. Are there any questions for the speaker? I have one question on the kimberlite that's radioactive.
Okay.
What are the radiochemistry products of the kimberlite?
Oh, so that's what I'll show you here. We've done analysis on that using SRF, and we got the composition of it to have this composition here, I've shown here. So we have CO2, water, phosphorus oxide, potassium oxide. So these are the composition of the kimberlite. And with the MIMPET process, our main concern is on the carbon and the oxygen, that is the CO2. So because diamond is full of carbon, that's why we irradiate the sample enough to irradiate the carbon content in the diamond, so that it can produce 511 KeV to be detected. Because if you check this chemistry part of the kimberlite process, you will see that the carbon has a mass of, if you do the percentage mass of carbon is 1.36%, and oxygen is almost 49%. And even I have another talk at Legon where I'm going to talk about the purely for carbon and oxygen on kimberlite rock. Thank you.
Okay, but when it decays with what, an alpha particle, beta particle? What does decay with?
Okay, so what we do is we bombard with gamma beam. So the reaction that goes in there is gamma going in, giving electron, and giving the unstable carbon 11. And then the end product should be boron, which determine the color of a diamond. So if it's blue, if the end product is boron, it means that the carbon, the diamond, the color will be blue. So the decay that come out is we use gamma neutron reaction. Hello.
Oh, thank you. Are there any other questions for the speaker?
Yes, my hand is up.
Oh, go ahead.
Yes. Well, thank you for the presentation. And I think you are basing your experiment more on the elemental composition. But I think if you had used X-rays, try to determine the structure of your material, that will have given a better result, because elemental analysis could lead to different materials, but X-rays can give you good diffraction peaks, which will tell a better story.
Okay.
Did you get that?
Yes, let me explain it to you.
Okay.
The X-ray components, like as I've stated here, is the X-ray fluorescence. That one was used to determine the chemical composition of the kimberlites. And during the irradiation, we use gamma source to irradiate the sample, because a diamond is very, very hard. So it needs gamma beam to produce the gamma neutron reaction. So there's a work that we've done on SRT, X-ray transmission tomography, with Gen Diamond was having that machine, and we did analysis for them. So when we use that SRT to examine the rock, we noticed that the attenuation was very, very high. So that's why we adjust to this MEMPET technique by using high energy beam. And in case if I want to use the same approach for gold, I might go for lower energy, because gold is not hard as diamond. Thank you.
All right, thank you.
Any other questions for the speaker? So hearing none, let's give the speaker a round of applause. It's a very nice presentation. We have scheduled to begin in 8 minutes the next speaker on Quantum Africa. Is the speaker here already? Faraya? If you're here.
Yes,
okay.
I'm here.
Let's take this 8 minutes to test your slides.
Okay, thank you.
And Gideon, if you could stop sharing, and then
Hello, Farai.
I'm trying to share my screen.
okay, you can share your screen now.
Hello, Farai. Good morning.
Yes, good morning, Professor Oga. How are you?
Okay, I'm here
All right.
to listen to you.
Okay, thank you. Thank you so much. Yeah, it's early morning. Let me try share my slides and see how that works. Thank you. I think they are coming through my slides now.
Yes, I can see them.
Okay.
So
Yeah.
we can let's for people that here. So your talk was supposed to begin at noon, which is 6 minutes from now. So we have time to take a tea break or whatever, and then we can start in 6 minutes.
Sounds good. Thank you.
5 minutes now. Thanks.
All right. It's good with me. Thank you.
Okay, Faraya, why don't we go ahead and get started?
Okay. Confirm you still have my slides on your screen.
I see your video, not your slides.
Don't see my slides. Okay. So let me see. What am I sharing here? Let's see.
Okay.
What about now?
Okay, now I can see your slides.
Okay. All right. Thank you so much for having me, and thank you so much for your patience. I know my speech presentation was supposed to be the other day, but got the confirmation late, and the time zone is interesting. So thank you so much for the flexibility to reschedule this and make this happen. So my name is Farai Majandu. I'm from Zimbabwe. Currently based in the United States. Joining you from the state of Louisiana. I'm a teacher. I'm a physics teacher. I'm a physics faithful. My undergrad is in applied physics from Zimbabwe. Then I went into teaching for 5 years, hoping to change how I learned physics and making physics perhaps more accessible to others. A goal that I think I achieved by making all my students pass physics by in 5 years by 2018. Then I left for South Africa, where I went to do a master's in physics at the University of the Witwatersrand in Johannesburg. That's where I started doing quantum information processing. So I did research in condensed matter physics, in exploring the applications of heavily boron doped diamonds in quantum information processing. And also hybrid quantum platforms based on NV centers in diamond and flux qubits, work which I explored using the IBM quantum computer that was made available on the cloud in 2016. And that is how I got into the field. I ended up participating in the QSKIT camp hackathon in 2019, as well as the summer school that was hosted at the University of Wits in the same year. And I was exploring how to use QSKIT, which is the quantum software stack for IBM quantum computers, but which also is agnostic now and connect to different hardware platforms. So I was exploring how QSKIT would be used to communicate with the quantum system other than a superconducting qubit-based platform. So that was interesting work, and that work and my team and myself a second place in that hackathon. And ever since, it has been my goal to see more people from the continent pursuing emerging technologies like quantum. And also, after having coming from a teaching background, I saw that as an important opportunity for young people across the continent. So there wasn't so much representation in the field, and that bothered me, and I had to do something about it. So I started building a community during COVID to just share my experience, share my research, expose that to more people, and become part of the emerging quantum computing community back then. So during COVID, I want to believe we built a very fast growing community that brought quantum experts from across the world to speak to the African community. We had people like Scott Aronson from the University of Texas at Austin. We had Bob Suter, then a vice president for IBM Quantum. We hosted an Africa Leadership Summit on quantum, which was sponsored by Honeywell then, now Continuum, and other players in the ecosystem. And I also started working as a consultant, started mapping the quantum ecosystem. So I started learning a lot about the ecosystem. So this is my journey that brings me here. Now I came to the United States soon after COVID to come to the Colorado School of Mines. I pursued another postgrad, another master's over there. Okay, sorry. I pursued another master's. I pursued another master's there. I'm trying to get back to my screen. I can't see it anymore. Just a moment. Let's see here. Okay. So I pursued another master's at the University of the Colorado School of Mines, and okay, I got my screen. Thank you for the patience. So I pursued another master's at the Colorado School of Mines, and now in the theory, quantum field theory, quantum physics, theoretical side of things, and started teaching here. And now I teach at a School of Science and Math, where we teach motivated students who want to go to college, to go to the big institutions, to do big science, and that's what I do now. So now I founded the Africa Quantum Consortium because of the need that I saw back then and the call from the community that we needed a platform for quantum leaders in Africa to come together and discuss about the future of quantum technologies on our continent and make that a reality for our scientists, our people who are coming up, and showcase the good work that come out of the continent. We nobody talks a lot about the research that comes out of the continent, or nobody seems to understand what's exactly happening in Africa. So standing on the shoulders of giants, who are the people who are convening us today, the Africa Light Source, the African Physical Society, and also thanking them for the foundation and starting building this foundation which we stand on, and also advocating for the International Year of Quantum, which is what we are celebrating this year, and which is what brings me here to talk about what are we doing in Africa, what should we do, what comes next, now that this is the International Year of Quantum declared by UNESCO and advocated for by these Pan-African organizations as well as other global players. So now let me just share with you the vision we have in the Africa Quantum Consortium. Is we are a neutral coordination platform for quantum efforts across the continent. We partner with those that have been in existence. We partner with those that are going to come in the future. We partner with those that exist, today. And our idea is Africa for too long has been fragmented, and that fragmentation is good for everybody else, but it's bad for the continent. We have seen how, despite having so much capacity and so much expertise from the continent, we haven't achieved much. Neither have we been able to put infrastructure on the ground. It's because we need to work together. We need to listen to each other. We need to share those goals, share common goals, and begin to put stakes on the ground. So we registered the Africa Quantum Consortium in South Africa. It is a non-profit organization with this structure that I share over here, and I'm going to show you some of the people that are involved. So I hope my slides are also transitioning as I go. So the Africa Quantum Consortium is a concept that I've been exploring from 2020 to 2021 with the Dr. Epistole, with Fiona Matu in Kenya, Dr. Epistole in South Africa, working for NISIS, and with Dr. Taha Roba from Algeria. And we thought over a long period of time how to put everything together, and what kind of a structure we could have, how we're just going to get started and be out there, begin to just get people to come in. What is our vision? What do we want to achieve, and how do we get there? So here you see the team that eventually crystallized out of this effort is what we have here, which we have the core team. I work with Dr. Taha Roba. He's a quantum tech expert from Algeria. He has been funded by the African Academy of Sciences. The Arise, he got an Arise fellowship. He runs what is called CQ Tech, Constantine Quantum Technologies Group in Algeria. At the University of Constantine one, and he's also a founder of a quantum startup, and his group has done very well. I work with Professor Ahmed Yunus from Egypt. He's one of the pioneers of the quantum computing field in Africa and within Egypt. I work with Temitope Adeni. She's a entrepreneur, a founder as well from Nigeria. Now she's based here in the US at the Cleveland State University. She's involved in quantum, and actually recently was at the Quantum Development Conference, IBM Quantum Developer Conference in Atlanta, where we even had, Africans who were showcasing their work over there and winning awards, which is really something that makes us super proud. I also work with Pumzile Madonsela from South Africa. I was in the same group with the University of Wits. She now works for FNB. Tadana Sogodam is from Ghana. Tokas Atuabia Ado from Ghana as well. Yusuf Traore from Cote d'Ivoire and Guinea shares, his background is shared in those countries. Omar Sobi from Egypt, and we work every day to make the consortium what it is. So I'm going to fly through this and just show you. These are the people that are involved in operations, and now let's come to the big story. We also fortunate to be advised by Professor Amadou Wage, who's here. He's also a coordinator representing us in Senegal. He's also one of our biggest supporters and sponsors, and we are super proud of the opportunity to have to stand on the shoulders of giant and being allowed this opportunity to take things further and drive impetus into the ecosystem. Professor Morad Teomin as well from Tunisia is also an advisor, and he helps us with the ideas and everything we need to move this conversation forward. So now the idea is we want to turn the International Year of Quantum 2025 as from the spotlight. We have the spotlight in the global south, but we want it to be a movement that goes beyond the year 2025. Because after all is said and done, I think we have had the year of light. Now we're having the year of quantum, and we want those to have tangible results on the ground to make impact, lasting impact as we go. So that is our focus at the consortium. And these are some of the things that we have achieved so far. So we started operating in earnest in November 2022. Then we officially registered the consortium as a non-profit organization in South Africa this August. But we had already, created a lot of momentum. So one thing that we do is we host leaders to come and share their work and share their vision, share their goals, and collaborate. So we have been bringing people together to come for the round table, the Africa Quantum Consortium round table. So we have run round tables, 3 round tables so far. Round table in the first quarter, round table in the second quarter, and round table in September. And we're going to run a round table end of year, which is focused on women in quantum from Africa and across Africa. So we have done that, and we have really been super successful. We have brought a lot of organizations on our platforms who are collaborating with us. We have spoken at global platforms, sharing Africa's goals, representing Africa, sharing all the good work that that's coming out of Africa. And recently we released the white paper that captures all that we are learning and the position of Africa in terms of the developments that are happening in the quantum ecosystem. Let's see here. Okay, so with those roundtables, we like I was saying, we have also gone across the world to speak at all the platforms we can think of. The Quantum World Congress in DC, the IEEE Quantum Week in New Mexico, Albuquerque, New Mexico. We went, like I was saying, even to the Quantum Developer Conference this year, hosted by IBM. We were at the ASFAP strategy presentation as well. We've contributed to that strategy, the quantum strategy part and quantum computing part. And we were represented by Oisao Momo from Morocco, and she's also working with Continuum. Now that we we have unified people. What else we want to do is to clarify what exactly does Africa need, where should we play, what should we do, and then we want to influence so that we represent Africa outside in standard, in standardization organizations like IEC. So we have a place over there. We are part of the World Economic Forum. We are part of ATTAC here in the US, which is a place where their government interacts, government agencies interact and think about quantum in terms of mission criticality and just how the public sector should be thinking about adopting quantum and also changing policy in order to enable quantum. So we contribute there as well, and we bring other organizations in Africa, like even Sakuti is going to be speaking at that platform and also connecting and learning and contributing. So that's our role over there, to make sure that Africa is on the table, is and also building the table with others, because that table for quantum is not yet there. Everybody is fixing it, and there's no reason why Africa shouldn't be doing that. So this we have contributed to even International Day of Quantum global events, the ones that are officially endorsed by UNESCO, which are hosted across the world. So we have participated in the Quantum Crossroads in New Zealand. We participated for the events hosted in Africa. We participated in Quantum Africa, hosted in Morocco. We have also started running grassroots projects in Zimbabwe, and our story is getting far and wide. And our recent roundtable was attended by 190 out of 290 people, 294 people showed up. We have introduced a quantum happy hour that the global community enjoys. We are going to roll out an Africa Quantum Hackathon. We call it the Pan-African Quantum Challenge. Our newsletter has been growing and keeps people, updated about quantum developments across the world and what's happening in Africa. And we released the white paper, which is now available. So the community is learning about what is happening in Africa, what are the plans we have on the continent, what is the best way forward from here. And we've also rolled out national coordinators to begin to have local representation in the country, so that we trickle down our ideas. We also, make it easy for people to contribute to our strategy and what we should be doing, because we can listen to people, deeper into the ecosystem. Because Africa is still predominantly a young continent with a lot of young people who have to come into the field, and we want to catch them young. We want to go to high school. We want them to know about quantum technologies, and we want them to get into modern physics, because when I was a teacher in Zimbabwe, I was teaching modern physics, and we did the photoelectric effect, and we could have, if ever quantum was a thing, then those are the places to introduce it. It can also introduce it in chemistry as a quantum mechanics, extension, and I know chemistry teachers teach those aspects as well. So it's very important to begin to talk about them. And computer science itself, even in high school, you can begin to talk about quantum computing. Linear algebra and mathematics is an opportunity to introduce quantum. And there are many other, touch points in the curriculum in high school where we can begin to do this. And then the idea is we want to train teachers as well. So we've got a grant that we've applied for with other partners, which we hope will be successful, and it will allow us to roll out, train teachers. So we'll start by training teachers in the 4 clusters in Africa: North, South, East, and West. And then these champion teachers, we hope they will train other teachers, and they will share some useful outputs with us, which are going to help us improve that program so that we can expand and improve it over time. So these are some of the key takeaways from the State of Quantum in Africa white paper. You see us here fleshing out how we will end up having a quantum computer in Africa. So the idea is we want to partner with the vendors like IBM, who have already been part of the ecosystem in Africa. They have given access to universities in Africa through ARUA. So WITS is in charge of that access pathway. And ARUA is also part of that. We want to bring also AAU, the other group of African universities, to be part of that project, so that they can get access to quantum computers. We're even talking to IBM about how to enhance that and expand it across Africa, and perhaps they have a much more broader framework of access. But beyond that, we want Africa to have its own quantum computer. The quantum computers today sound expensive, and everybody is saying, no, Africa can't have quantum hardware. We think differently. We can't solve the problem of tomorrow by looking at the hardware of today, because quantum computers are going to change. The stack is going to be figured out. Some of the components are going to even go away. We may not even need them as new ideas emerge. So nobody imagined that a computer could end up fitting in your hand as a cell phone. Right now we've got smartphones, powerful computers. But imagine if you thought about the computer, the big, computers which were big as warehouses that started back then, nobody would ever imagine we'll get there. So innovation will happen. So we are not planning for a quantum computer of today and thinking that the hardware will always be expensive. And after all, recently China proved that it is not the case. Pakistan has been ordering a quantum computer from China. Just a couple of millions. It's ion trap based. It's just a couple of millions, and we see that happening. And we've even seen it in AI, where people have been training these huge language models, and the DeepSeek moment with China coming up with a lean large language model, which has been doing amazing. And I think there's been a lot. There's a lot that's happening, and we'll see that shaping as well the same way in quantum. That's our vision. So that's our belief, and we think we are quite, supported by history in thinking that way. So Africa should end up having its own quantum computer, having its own sovereign infrastructure, just like the light sources and all the infrastructure that all of us here are advocating for. So that's our vision as well. Then we also created this, ecosystem flow diagram to just show you, to say what is the governance like for the quantum ecosystem. If the African Union could have, a section within the organization which begins to champion this Pan-African quantum strategy and concept, and the situation room within the African Union to think critically about the developments that are shaping our world, that, including quantum, including AI, including space. Because these are huge undertakings that need a whole continent to get behind, and African nations have to begin to think differently about research, about building capacity. And even the governance models have to shift, and we have to start funding our own infrastructure, honestly. It's not sustainable that we continue having our infrastructure being, piggybacked by somebody else. And we hope these are the, these are the thoughts that are beginning to grip our government. And through people here and through other platforms, we hope this message is going home. We sent this message home at the ICTP symposium through the report in the strategy that we propose, and we're doing the same here, and we'll continue doing the same. We are engaged with, we are reaching out and in conversation with the Order NEPAD and the higher, I think they call the emerging technologies section within the AU, so that they know what's happening and they should think critically about what to do. I hope the geopolitical shifts are forcing leaders in Africa to think differently about how to invest in their own continent and make things happen. And that's our big call to them as well. All right, so we thought even deeper. And let me go back to this slide. We thought even deeper and came up with a quantum compact. We're going to release this document soon, which shows how 100 million invested in Africa over 15 years is going to make the continent, quantum secure. We have our own sovereign infrastructure on the continent. We have an ecosystem, a quantum ecosystem on the continent. We have a scientific ecosystem. We will be able to generate the capacity in terms of human capacity. We'll be able to generate the startups. We'll be able to fund them. We'll be having governance structures. We have the whole stack. And we'll also have our diaspora network being able to contribute through what we call the Quantum Homecoming Fellowship. And with that 100 million, we'll be able to take the continent into a new era, never look back, and change the trajectory and the future of our continent. Also able to achieve our Agenda 2063, be able to deploy all the tools that are available, the best tools that will be available at that time, to secure our connections, to build socioeconomic capacity and development that makes people proud to be African, to beneficiate our resources, to solve our biggest problems, eliminate poverty, deal with health care, and set the continent towards that path of prosperity that we all desire. So that's our vision. That's some of the ideas that we have, and this is who we are. This is the Africa Quantum Consortium. This is what we work on every day. So if you scan the QR code, you'll be able to go to our website and see, find ways forward in terms of our presence on LinkedIn, in terms of our upcoming initiatives, and in terms of, just being part of the ecosystem and begin to also contribute and learn with us and grow with us and build with us. And I leave you here with a quote: it is within the possibility of science and technology to make even the Sahara bloom. Not one of us working singly and individually can attain the fullest development. This was Kwame Nkrumah at the OAU inaugural address on the 25th of May 1963. So we hope you join us to secure the future of Africa together. And thank you so much for having me.
Okay, thank you. Are there any questions for the speaker?
And I will also be able, I will be sharing I see it here, how can I be a member of your group? Sure, I will share a link here shortly when I get off stage, and you'll be able to, find where we at and begin to engage, and we'll be super happy to work with you on what we are doing. We are aligned hip to hip. You guys have the foundational science and the infrastructure concept, and we are now just taking it to quantum. But otherwise, it is the same physics, the same scientific infrastructure that we need to get there. Thank you.
Lawrence, can I say something?
Sure.
Thank you. Thank you, Farai, for your talk. It was very interesting, and we will learn a lot about the Africa Quantum Consortium. Thank you very much for giving this talk at the African Physical Society and African Light Source joint meeting. Thank you for taking your time for that.
I appreciate it. Thank you for your support. Thank you for your advocacy. Thank you for all the history that you have, safeguarded for us. And we are proud to stand on this strong foundation you have built for us to go further.
I have a question.
Sure.
When you use the word quantum in this context, what do you mean in terms of mapping that back to how physicists are trained at the undergraduate level, at the graduate level? So we've all had experiences with our graduate quantum class. And maybe some of us have had experiences with quantum chemistry. Now for years now, there's been the coming on of quantum biology. In electrical engineering, there may or may not be a course in applied quantum mechanics. What do you see as sort of driving this sort of popular use of the word quantum?
Thank you. So there is the foundational side of it, where we are not really talking about the ability to control the individual particles, electrons, photons in the full flavor of the laws of quantum mechanics, taking advantage of superposition, entanglement, quantum interference, like that. So because the moment you come into the community and you want to introduce something totally new, it's very hard to get adoption. And it's also important to mention that they are just levels to quantum, and now we are at that highest level. So when we are in your high schools and stuff, we talk about the photoelectric effect and just the breakdown of classical physics and introducing modern physics. Then when you get into quantum chemistry class, we now introduce the fuzziness of, like, for example, you start by teaching, normally we start with that model of an atom which has got well-defined orbitals, and the electrons are particles. So students over years imagine electrons to really be particles, fully self-contained and localized. And then you begin to break that down, saying this is why we introduce electron clouds, and then that makes that whole chemistry gives that full richness of chemistry. Then if you talking to physicists, depending with their level, after the breakdown of the classical physics side to quantum, then perhaps it's now more going further to the photon level, and depending on the level, going further even into the quantum field theory level, where you begin to think of fields and a wave particle duality in its full flavor. How maybe thinking about fields and ripples of fields as particles, and how this is just a matter of convenience, and depending on where it suits the context. Then for electrical engineers, it's more of the engineering side, how to make devices. So maybe the high frequency side of things, your microwaves, your waveguides, and making those smaller and taking them into the quantum hardware and enabling, building technological platforms, which we are able to do now to build the quantum computers and the sensors and the communication links that people are exploring. I don't know if that answers your question, but these are the different ways that I'll be thinking about quantum every other time. So it just depends. Who am I talking to? What is accessible to them? Should I use the nuclear magnetic resonance for MRI so that I show them it's been happening? Should I use the energy levels in materials, like what brings us the semiconductors, so that is accessible? Or do I go further to show them where we are today and what we can do today, and why we are able to do more enhanced things in information processing, in sensing, in communication, in terms of secure communication and things like that.
Okay, thank you. Are there any other questions for the speaker?
Just okay.
Jeff, go ahead.
No, it's okay. I want to talk to you after.
Okay. Then if there's no other questions for the speaker, then let's give him a round of applause.
Thank you very much.
And thumbs up. And then so we're scheduled to go on a break from until 1400, right?
So Lawrence,
what time is it now? Yes.
Just before going to break, we have to go back to the ICTP meeting. And
right.
And I know that Omolulu is not in, so
he just sent me a message, said that he can join.
Yes, that he's at the airport. So if you can do it.
I guess I can.
It is for 15 minutes, and after a discussion. And me, I just make a small introduction, thanking all the contributors and so on.
Okay.
I'm going to talk just for 2 or 3 minutes.
That's fine.
Okay, thank you.
So we are on break until 1400, which is basically an hour and a half from now. Right? Yeah. 2 p.m. GMT plus 2. And the talk at that time will be from Dr. Make sure I get it right. Right? Kersey Lawrence from the Cypress Institute. And the talk will be on bioarchaeology and her physical methods used therein. Okay, so let's see. Okay, so yeah, we'll break for 1 hour and a half.
People are at ICTP, they are not ready for us.
Needed speed to get a funding to build the FCC. If the FCC is approved, then China will join the FCC. TV, I think you're taking that. So I think let us thank Professor again for the presentation. Thank you. Now we'll go into societal engagements, and it's going to be convened by Mark. Mark, he's online. Professor Wanye.
Yes. Yes, I am online. Yes. Do you hear me?
Please go ahead. It's your session now.
Okay.
Yes, we can hear you.
Okay, thank you very much. And first of all, I would like to thank all the contributors to the African strategy for fundamental physics and applied physics. And especially, I want to congratulate Ketevi and all the other people who have contributed to this great work. And of course, it is a great pleasure for African Physical Society to be here today with you, because from the starting, African Physical Society and African Light Source are the first organization to give floor to African strategy for fundamental and applied physics. And now it is a pleasure to see that the report is ready. And I hope that this report will give a new will be a new step for the development of physical science in Africa, and also that it will bring many sponsors and African governments to understand at least that science is important and that they have to give money for science and have to support scientific institution and scientific society like African Physical Society and others who are doing what they can and sometimes without almost nothing. And for that, I would like to thank ICTP for constant support to African Physical Society. And now I will give a floor to Lawrence Norris, who is going to present African Physical Society and maybe also to have a feedback, because Omonulu, who is supposed to talk, I think is at the airport for the moment. So thank you very much for the organizer. Thank you again, and congratulations again to all the contributors in writing this report. Thank you. No, Omolulu is not here, but Lawrence. Oh, I am sorry. So Omolulu, you are in?
I thank you for having me. I'm sorry for the situation. I'm actually traveling. I just landed at an airport. So I'll present it from there. If there is no too much for me, like the announcement, I'll but I'll share my screen now. Can you see my screen now?
Yes.
Yes, we can. Okay, thank you very much. So I'll be discussing about our feedback from the society. So Professor Ahmad Bagi, speaking the society. Lawrence Norris has been very active as part of the executive council and I'm also not. So I'll tell you in 2 76 here about Morocco. So if you want, you can date AfPS system to 2010. So tell you a little bit about the vision for AfPS is to enhance the timely development of physics education, research, training, and both national and transnational collaborations for the benefit of African and global physics community. In more details, give the vision there is to enhance the development. The vision is to serve as the continental voice for physics, providing a forum for networking collaboration and uniting national. So this one just shows the northern region, and then we have the Central African region, there's the Eastern region, which is called the Southern Africa region. So at a glance, there are more than 20 national member societies that are part of AfPS, and we have it as the Pan-African network. If you go to the website, the list of the 20 national member societies that were created, or associations that were created by AfPS, is the African Association of Physics Students. This is very important because of course, we are going to the students are the ones that are going to continue the association and continue the work. So AfPS created AAPS, African Association of Physics Students, to encourage physics students in their scientific and professional work and things like that. And then there's also the African Physics Teacher Association that was created by AfPS. Now AfPS is trying to work on creating a federation of Pan-African physics societies, which include AfPS, APTA, the National African Astronomical Society, the Society of African Physics Societies, Christian Association, and also the African Material Research Society and other societies. So AfPS is the integration of Pan-African physics societies. So the main topic that we have today, AfPS. So first I'll talk about the fact that the ASFA received a mandate from AfPS, African Physical Society. I'm not sure exactly which year. Professor Wage will be able to tell us exactly which year. And the mandate was to develop a strategy, African strategy for fundamental and applied physics, and develop it as a community study to define a roadmap of physics research and education in Africa. Some of the statistics I've been able to get have been about the ASFAP and how it was made. It's what I'm showing here. So ASFAP is a product of about 400 African researchers, representing a community of over 1,000 from about 42 countries. The breakdown is as shown here. So from the one of the reports, claim that they work very hard to the 60 thematic working groups. The core contributors were about 400 African physicists. Okay? And there's a wider community. So this 400 African physicists communicated with other physicists together. So roughly this will come to like 1,000 physicists and from about 42 countries. And then there was also international support. So it's a big, I'm sure we already discussed it. So it's a big effort and to create the strategic plan. And the summary of the strategic plan the strategic plan that was produced can be summarized 8 recommendations. The first recommendation is actually for future strategic planning, and then the remaining 7 are recommendation for development of physics system. So, so our first feedback is to say a big congratulations to Dr. KTV and the whole group that worked on the ASFA. So this it took a lot of time, so and effort and energy. And as Professor again, the AfPS president said, we congratulate Dr. KTV and the whole team for this very big achievement. So this is a big thing. I think it's the first time this has been done in Africa, and also to do it to this depth and to this level, and to make it this broad is very big achievement. One feedback that I would like to give would be to say, perhaps one of the things we should include is something about performance metrics. Okay. So this is a suggestion. There may be pros and cons for why or why not. Why you should have a performance metrics or why you should not. But it would be helpful, for instance, to talk about in this strategic plan, our goal is to make sure that there's a so-and-so percentage increase in total GDP contribution to physics society. I know it has been, this kind of performance metric has been used over time, and sometimes it works, most times it really doesn't work. The countries don't and may not be concerned. But I think at least we should try to have a target like this, or a performance metric talking about that are trained in Africa. So this will be something that I think would be good to add general to the strategic plan. This is already in the strategic plan, and I think it should be a main focus on capacity at all levels. This is one thing that is very deficient in Africa. Living in Africa now, I see that this is Professor Wagi will also agree that we need a lot of capacity at all levels, in primary schools, tertiary institutions, and also for the case of kids. A metric could be like 4 times the number of kids, 8 years. At the primary school level, yes, many of us could say that maybe we don't physics in primary school, but the thematic community and outreach is very vital in this area, where the community simulates young minds to think about astronomy. Beginning, they're already planning to engage in physics. So at all levels, including primary school, secondary school, I think it's important for us to. Now, one of the things I saw that was noticeably AI or machine learning in all the different every thematic groups, even if you are not directly involved in AI or machine learning, it would be good to get it. It's hard to imagine the possible future that AI is not possible today. Even in climate modeling, of course, some people already discovered that to help in the modeling, and it's not just the phenomenological equation that you have, but AI can help to make the modeling faster. So I think in every area to incorporate AI for machine learning in all the thematic areas that we work in. Nowadays, of course, there are people that are already including machine learning in this mathematics. So it's not just traditional mathematics. There's a need to show. I think this will and I want to piggyback again to say perhaps we should also think about focusing on capacity change. So here is the UNESCO report. The number of changes belonging habitats in each country and in each region. Yeah. So you have people's months. So the one average is 1368. This is the 28th because the figure in brackets is in 2014. So the one average is this. Unfortunately, sub-Saharan Africa, which excludes South Africa, has an average which is like less than 10 times one average, both in 2014 and 2018. So I think what's important thing is to build capacity, focus on capacity building of PhDs. Europe has like 3 times one average. I think Japan has the highest, and followed by Canada, US, Europe, and other parts of the world. So I think it would be good if this would be one of our purpose to build capacity in PhDs. The final feedback we want to give is the one we started, and is to say congratulations, very big congratulations to everybody that worked on ASFAP, including also the Dr. Thank you. And with this, I would like to thank you for your attention and end my presentation here. Thank you very much. Yeah. I'm going to take questions.
Thank you very much, Omoduru. It is a great talk. Thank you. Now we can open question up to 11.
What?
If some people have question.
Okay, yeah. I can answer the question together with Prof. Again. I have a comment. Yes. I would like to Professor Wanye and also Professor so for the So, thank you for so I really appreciate it. And I look forward to continuing to work with you for the benefits of our country. Thank you.
Thank you very much, Professor Ketevi. In our congratulations, we have also to congratulate Lawrence Norris, who have done a lot of work on behalf of African Physical Society. Thank you, Norris. And again, Ketevi, we are together, and we are going to continue to work together to see how to implement this report and how the African government can take in charge of all the recommendations which are inside the report. And we hope that this time there will be a positive answer from government in Africa, because it is important that we need local funding for the development of physics in Africa. We cannot always wait for help from other countries or for international organizations. Of course, they are very important. And again, I want to congratulate ICTP and UNESCO for what they have done to make this open symposium of ASPA to take. So thank you very much to everybody. Thank you for all the stakeholders. Thank you again, Ketevi, for your tremendous work. And we hope that in the future, Africa will go ahead, because we have a very, very smart people. We are the youngest continent in the world in terms of population. And I am sure that with all these positive people, we can go ahead. Thank you again. Omolulu, or maybe Lawrence, you have to say something?
Yeah, thanks a lot for this very nice presentation. Nice to see. I think this is a great initiative which can be very important for physics on the continent. I have not heard much about it, unfortunately, and I just have one comment. If you get to your website, it would be nice if you could also link it in the slides. Then my question is, who can become a member and what are the requirements? So that's my first question. And then my second question on slide 5, did I understand correctly that it's only around 20 national societies in Africa that are affiliated to AfPS? Is that correct?
Yes, to the second question.
Okay, so that's less than half of what the state of the continent, because it's around 54. So what are you doing to increase your visibility and get to know more people joining? Because I think this is a great effort.
Yeah, I think important is to realize that many African countries don't have viable physical society. So Nigeria has one, some other countries have one. So it's not like every African country has a physical society and some are neglected. The idea is that the issue that many don't people that have a physical society. That Professor Wage can say more about this.
Yes, you are right. The main problem is that physical society that exists in many African countries. And one of the main goals of the African Physical Society is to help to create physical society in all the African countries. And we are doing this job, but okay, for that we need also to the development of physics, the capacity building, because in many countries in Africa there is not a lot of physicists. So at the PhD level, this is why Omolulu says that the capacity building is a key element for Africa. Thank you. So,
can I come in there?
yes, yes. Thank you again to everybody.
I want to thank Sylvia for your question. You are a young person, energy, and I'm online and in the room. We have a lot of young people with their young energy. And often when you are an emerging researcher, you're very concentrated on your research. But the problem is vital. You take some time to concentrate on the health of your discipline. No one is going to do it if you don't. Everyone must take on an additional burden beside their research to progress the discipline, to make sure the discipline is healthy. I want to really tell you, if you don't do it, don't be surprised if it doesn't happen. And something like this African strategy or any kind of work in service of the discipline is an example. You make the discipline better for the continent will rise. The science is an engine for development and peace and democracy and wealth, all of these things. So really want to say, just go to the website. You don't even need the link. I know, James, that you find a way. Please go there and just register yourself in the African Physical Society and start to work, even if you have to create any subdivision for some kind of task that you see. Just please take the initiative. Same in your own country. If there is not yet a physical society there, there's nothing to stop you making it. As you heard how the foundations of the African Physical Society were vision. Just get together and decide to make it. It's my comment here. Specifically target the young people. You have to absolutely get involved in these things. When a government looks at the landscape and they see the societies, these are voluntary associations, they professional bodies, they are not statutory. It's self-organization within the community. When they look at that and they see a strong membership, that's when they are going to take heed. They need to see the numbers, they need to see the activity. Sylvia, also especially to you, all of your cohort that are with you, please get involved. Thank you.
Thank you very much, Simon, for this nice comment. I have to add that Simon is one of the great leader in Africa, and also working for African Light Source and African Physical Society. And every year we are organizing joint meeting with African Light Source. And next year in Zimbabwe, there will be an in-person meeting. And Professor Simon is very, very instrumental to all the scientific activities in Africa. Thank you, Simon, for your support and for your energy.
I know something will have as his virtual hand raised, and then goes to ask a question. and I think they are online or in the room. So who are willing to say something, but we need to take another snapshot of all the delegates. We want to do that now. So please start to turn on your cameras. And while you're doing that, I want to remind you, those who are here in person, just at the tea break, we have the group photo. So please be ready, see where it is exactly, and we'll have it. And Ketevi, maybe tell us where it is, if you know. No, I think that once we are done here, she will tell where to go to take the photo. Okay. Okay. Meanwhile, everyone, you should be turning on your camera and maintaining a smile. I apologize. My camera is very close from here, so you don't want to take my snapshot right now.
I turn on my camera and I am smiling.
Okay. I don't see a lot of cameras. We should have some more. I'm starting the snapshot. Okay, are you ready? Smiling. 3, 2, 1. That's the first page. I'm going to the second page. And again, 3, 2, 1. And I take the snapshot. Thank you very much. That's both snapshots. Everyone, we break now for lunch. Be back here at 2 o'clock. Thank you.
Thank you, Simon. Thank you.
Hi, Kirsty. This is Lawrence. Thanks. A quick question. So we usually record for, we're trying to make this video library, which is a whole different story. But is it okay when we record your talk?
Yes, absolutely.
Okay.
Can you hear me?
Yes.
Ah, wonderful. Okay. And
Right.
yeah, absolutely. I adjusted the slides so that they.
Okay. So I have so about the video library. I have hours and hours of video of talks that I'm supposed to do something with that
Uh-huh.
I have done nothing with.
Bon courage.
Yeah, yeah.
The day will come. The day will come.
The day will come.
The day.
There's a service that Springer Nature has started that so if you have a recording of a lecture, you can upload it to the server and the speaker can upload like supplementary materials. Now, so either you can post-process the video yourself or pay Springer to do it. So it's like the video equivalent of a page charge.
Right.
Right. So I have many hours, and so the price gets up there.
We'll need to find a funding body.
Yeah, I think I'm looking for a 15-year-old because it's simple work. It's just tedious.
Yes, absolutely. Absolutely.
So
But would be a good experience for a certain stage. Yeah.
yeah, there's a video. All it is somebody who knows how to use a video editor.
Exactly. So just to double check, you can see my slide,
Yes.
title slide in the full presentation mode. That's great. And we'll do the 25 minutes plus 5 Q&A, right?
Correct.
Wonderful. Superb. So yeah, I think it's a challenge to get all of this dissemination out in the end. So maybe in the future they will have an AI assisted edit mode where you just tell the AI assistant to do it.
Yeah, that would be great if there could be an AI video person. I wonder if there probably is that actually.
Yeah.
This could cost just as much money as a 15-year-old.
Yeah, that's right. Maybe a call out through the lamp for school project. You know what I mean?
Yeah, sure. I mean, there could be
Interesting
somebody,
science project.
yeah, who's can make a digital library.
Yeah.
Maybe an undergraduate student in information sciences.
Yeah. We're brainstorming. We're coming up on opportunities for.
Yeah, right.
Very good.
But it probably just comes down to I need to just make the time to clip the videos myself. It takes maybe 5 minutes per video, but it would take me the number of videos I have.
How many do you have?
It'd probably take the greater part of 2 days.
Oh dear, you're getting into the triple digits, I think.
Yeah, sure.
Oh, very good. So you'll be chairing, right?
I will be.
Okay. Do you get questions on the online?
Yeah, so what I usually do, so I'm not that great of a session chair because I pay more attention to the talk than the clock.
Yeah,
So,
I'll do the clock. Don't worry.
So what I do is this is open up the questions. People can unmute themselves and ask. And sometimes people just put the questions in the chat and I will monitor the chat and we'll just go from there.
Okay, perfect. And just to reiterate, I'll need to hop into the car at 2:30 so that I will my next meeting. So I'll keep it to 25 minutes as a talk and then 5 minutes questions. And I'm happy to answer afterwards online on the chat or whatever if there's further questions. Would that work?
Yes.
Yeah.
So why don't you just go ahead and get started?
Wonderful. Okay.
Do a quick introduction. Kirsty Lawrence from Cypress Institute, who's going to talk to us now. Go ahead. That was short and sweet.
Very much. It's a real pleasure to be here today. with all of you at the joint African Light Source and African Physical Society Conference of 2025. Thank you for the organizers. Thank you for your kind invitation. And thank you, the audience, for joining us today for this talk. What I'd like to take you through in the next 25 minutes, followed by your questions, is looking at how synchrotron radiation-enabled advances in human bioarchaeology are currently developing, looking at current research and future potential, but also making it hopefully directly relevant to you in that I'm looking at the beginnings of the application of synchrotron radiation-enabled approaches to a particular scientific domain where it started relatively recently, how synchrotron radiation approaches enable us to address key questions of wider relevance, including within the framework of the UN Sustainable Development Goals. And in so doing, also referring to perhaps some opportunities and some developments that might be of interest for the African continent and in the context of synchrotron-enabled research, as well as opening up some collaboration opportunities for those of you who'd be interested in contacting me further about those. So let's launch into it without further ado, and I look forward to your questions in due course. So synchrotrons, here's an example of one. Sesame, it's located in Jordan, but it's our very own synchrotron as Cyprus as well. We are one of the members of Sesame Synchrotron. And Sesame is special in this talk's context as well, because this facility opened its doors with the first user beam time on human bioarchaeology, research on archaeological human remains. So Sesame is a good example to focus on here. The beam time in question had a very international participation, and I'm very pleased that there was a foothold there also from the African continent, from Egypt, the gentleman there on the right of the standing team members that I was able to take to Sesame at the time, Yosef Hafez. And then, of course, we had the pleasure of collaborating with Masoud Harfouch, the beamline scientist from the XAFS XRF beamline at Sesame. And one of the things, of course, with this type of work is we need to start from the basics. I assume many of you may have not come across human bioarchaeology as a domain of scientific activity prior, so a very brief introduction. It concerns the study of human remains from archaeological contexts, and it emphasizes really the integrative, interdisciplinary analysis of links between biology and culture in past societies. And the beauty of this domain is that many different synchrotron techniques and approaches are applicable. We don't just use one single technique or single type of beamline, but many, and are very interested in multimodal analysis as well as correlative multimodal analysis. We want to know about the structure, the shape. We want to know about the chemistry. Here's a taster on what's been happening. I mentioned that the uptake of synchrotron approaches in the domain of human bioarchaeology has been relatively slow compared to perhaps some other domains. We have the first publication on human teeth from 2000, so only 25 years ago. And from there on, a trickle of publications coming through, and finally now starting to take off with a few more per year. The uptake was earlier in cultural heritage domain in general. But why should you care about this particular domain and synchrotron use in human bioarchaeology, its history, challenges, and potential? Because they highlight the need in general, when we look at new domains of application, for user community building, for the sustainability of such large infrastructures. And I'm happy to report that we have a user community building workshop taking place coming Monday and Tuesday here in Cyprus, funded by one of the EU projects in support of Sesame Synchrotron, the Sandstone project. And such user community building, including in the domain of life sciences, that I have also engaged in within the African continent in the framework of the Comelis project, and particularly Comelis Globe, is very important in reaching out and diversifying the user community. And we also can engage from our respective research domains in infrastructure development. I will give you some examples of feeder facility development, national laboratories that can help users both nationally and regionally to take advantage of the synchrotrons that exist in their region as well as globally. And the regional and transnational collaboration are, of course, aspects that are important in this context. On another note, in terms of the UN Sustainable Development Goals, of course, when we look at human health, whether it is in the past or present, and in my case, particularly human metal exposure in the past and in the present, this has resonance with a number of the UN Sustainable Development Goals, ranging from good health and well-being to gender equality, clean water, decent work and economic growth, and so on and so forth. The importance, of course, that is brought forth by focusing on a particular scientific domain like human bioarchaeology in the context of synchrotron use is also how it can illustrate how important synchrotron facilities are in enabling us to ask new research questions with real impact in our current world, as well as resolving those long-standing questions in a more efficient, in a more detailed way, if you like. So this is what we'll do in the next while. We'll look at some of the key research questions, and I like to always put it to my students and collaborators when we are designing research projects. A question that is of importance is, what is the impact? What is the ultimate impact of the research results that we hope to obtain? Now, there are obviously some challenges on the way in terms of the application of synchrotron radiation approaches to archaeology. Archaeology, perhaps in many minds, is about digging in the dirt, and indeed that's where it starts, recovering the material that is then painstakingly recorded and taken to laboratory context to analyze. And ultimately, in some cases, samples are taken to synchrotron facilities, in some cases in vacuum conditions in terms of the sample chambers. So really from dirt to vacuum, if you like, and there are many challenges on the way. I'll show some case studies, a couple of them, to give you a flavor what is currently being done, and hopefully to inspire also some questions and some discussion, and perhaps even inspire your own work in your particular domains with your particular samples. And we'll also look at some future prospects. So a very quick introduction, mentioned already, it's human remains from archaeological sites. What we hope to find out is about people's lifestyle, health and disease, their cultural practices, as well as mortuary practices. So just stressing that it's not just about the moment of death and after, not just about mortuary practices, it's about life. How did our ancestors live? And there's different scales of analysis, from population level to individual level, all the way boring down to tissue level. And these remains are our most direct avenue to access information on past people, our ancestors. So bioarchaeological samples, of course, in a wider context can include any of these, but we'll focus on the human remains today. And as mentioned, here's some of the publication history. It is a relatively short, couple of decades long effort so far, as opposed to the uptake of some other, at the time, cutting-edge novel approaches. Here, as an example of radiography, and that in its uptake in terms of human bioarchaeological inquiry was extremely rapid. So we have the first radiographs as showcased here, and within the same year, a publication came out with radiographs of archaeological human remains. So really, really rapid uptake. So the question is, and this is what we're exploring, what has been the underlying causes of the delay in uptake? Here, looking at the synchrotron beamlines used so far for human bioarchaeological research, and to note, Sesame has the core techniques for human bioarchaeology and cultural heritage applications. And I'd like to thank my colleagues very warmly for their collaboration, Masoud Harfouch, Jihan Kamel, and many others at Sesame. Here's its location. So I believe this is the closest synchrotron to the African continent, and many scholars have already come from the African continent. Here's a schematic overview. And here's the current beamlines, and we are happy to be inaugurating on 8th of December the AfPS beamline. And there has been some discussions in various quarters on the future. And as devil's advocate, I would just like to drop the note on what about an African beamline? Andrea Lausi, the scientific director of Sesame, would certainly be very interested in hearing from you on that front. And these type of beamlines, of course, can give multiple advantages in the journey towards one's own synchrotron, so to say, the Australian synchrotron story being a case that can be looked at, its first involvement with KEK in Japan, and then a road leading to a full-blown construction of a specific synchrotron facility. So let's look at Sesame and the human metal exposure work there, as well as some of the other questions that can be addressed through use of synchrotron radiation. We can look at any of these: preservation status, analysis of tissue structure and alterations, age at death estimation, looking at dental data, osteobiographical reconstructions. This pertains to information on people's lives, what kind of pathologies they may have experienced, and so on and so forth. And metal exposure may be part of that life story, if you like. Dietary reconstruction, medical treatment techniques, and so on. And the key thing is to consider what are the key questions that can be answered. And this is also, of course, what gives the best success in the beam time proposal stage. Now, let's look at some of the challenges. I mentioned that there are some on the way from excavation via lab to the synchrotron. And no single person today is expert in all of these steps. So it really requires teamwork. And I think that is also one really wonderful thing about this type of work. We get to work with many different experts from different domains. In addition to this, sometimes it's necessary even to construct new national strategic infrastructures. And here's what we were able to do in Cyprus, lest it would inspire some thought in terms of construction of feeder facilities, and national laboratory facilities, strategic facilities on the road for the African synchrotron light source. So here's a grant that I was able to secure from our national funding body within the domain of life sciences. We think out of the box, although I mainly do archaeological work. Given that we are focusing on human remains, I was able to hit funding call that focused on life science, and we were able to secure substantial funding for building of a infrastructure, a platform that has in its heart a micro-CT, a walk-in hatch, custom-designed micro-CT. And here's a picture of that with my colleague Anis Fatima, who effectively works as a beamline scientist would. And indeed, prior to her coming to Cyprus, me being able to recruit her, she was working at the Indian synchrotron at the SXCT beamline there. So such facilities have multiple advantages. We can, of course, do research in its own right, but we can also assist users in terms of their road to the synchrotron. And indeed, we are a sister facility and twinning with the Sesame SXCT beamline. And most recently, we're now building with a new grant a chemical imaging, chemical mapping facility that is involving instrumentation partially integrated into the existing custom designed open micro-CT walk-in hatch. So we will have XRF capability, XRF mapping, as well as a lab XAFs for chemical speciation. So feel free to contact me for further information or collaboration on that. So that as an example of how we start building feeder facilities that match beamlines at the synchrotron facility. Now we have two of those matching the SXCT tomography beamline at Sesame, as well as the XAFs XRF beamline. And next in our dreams is a infrared facility. So challenges in particular in terms of looking at human bioarchaeology, but in many domains indeed, with synchrotron radiation-enabled approaches. Some of them involve identifying the right approach. So that means discussions with experts from the ideation stage all the way to research design and submission of the beam time proposal. And for this, nodes at national level, as I showcased, national strategic labs connected to synchrotrons can be key, or regional ones. prior laboratory analysis are indeed critical for beam time success in the proposal phase, and accessing appropriate facilities is sometimes challenging. Sometimes we need to build them. So sample preparation and relevant facilities can also be a challenge. And then once we have had the beam time, sometimes the data analysis is the challenge. Analysis, interpretation, and then dissemination, they are, of course, a key deliverables afterwards, but data analysis may require advanced expertise and having national or regional contact points and resources, human resources to draw on is a key. So then acquisition of representative number of data points. This can be sometimes a challenge in terms of limited beam time. So it's not enough to showcase proof of concept, quest for the firsts, and then that's it. We do need a number that is representative, and that sometimes takes quite a while. But the good news is, in this community, I feel that we've been really exploring frontiers together, pushing the limits of techniques whilst exploring and identifying new research questions. And heterogeneous samples are, of course, the ultimate challenge in archaeology. So correlated multimodal imaging and some of the developments, looking into sample holders in different beamlines and how we can deal with that side of things. Okay, so let's look at briefly a couple of case studies. First one from the Sesame synchrotron, and here we are focusing on the preservation of human hair, comparing it to modern hair. And I had the pleasure of working with my colleagues Ali Metin Biyukarakaya, who's the archaeologist, bioarchaeologist in this one, and Jihan Kamel, the beamline scientist from Sesame, evaluating the preservation of organic compounds in this particular material. Some images of the context: the site is flooded. Some of the experimental parameters here. We are at the beamline in Sesame, and a brief look at results. And the key result is that we are certain that there are potential for further analysis with techniques relying on organic compound preservation that in many cases are destructive. So we wanted to make sure of the potential before launching into it. So you can read more about this study here. And here's the acknowledgement. So always a big team, of course, and many funding sources. Let's look at metal exposure. And here I'd like to very much thank my colleagues from ESRF, from ID21 Beamline, Marin Gott and Wout de Nolf, as well as my Iranian colleagues listed there, and my then PhD student, Grigoria. So here's the context: very arid domain, and Farshad Foruzanfar, my bioarchaeologist colleague, and we are there recovering the material in the desert near the Afghan-Pakistan border area of Iran. And here's the publication where you can follow this up. Here are some of the hair remains, and I thought I'd also put this mirror in its wooden box to just showcase the absolutely fantastic preservation of this over 5,000-year-old material from the site. And copper production was extensive in this region, and we have later sources that describe also the strong fumes and expectation of harmfulness to human health. What I wanted to look at is looking at the biogenic versus diagenic genetic uptake of metals. So what comes from the environmental contamination after burial, and what comes from the actual biological uptake. Here's some of the materials and methods and some images from the effectively we mapped thin sections, about 10 micrometers of hair strands. And you can see, if you can see my arrow there on the right, there are cross sections of hairs from different individuals. And this particular individual in the top left shows much, much higher copper levels than the other samples localized in areas that are consistent with the hypothesis that it is biogenic uptake. And then, of course, it comes to the interpretation. All of the individuals, they had low levels or background levels of exposure, somewhat higher than usual. But one young female had particularly high levels. Was she a craftswoman? Did she use particular different kind of cosmetics from others? And so on and so forth. And this, of course, has relevance to critical health issues today. We all use mobile phones. Just think of what metals are there and how they are sourced. And World Health Organization highlights human metal exposure as one of our challenges, key challenges of our time. The acknowledgements. Let's look at future prospects. Where do we go from here? So we need to address those challenges that I addressed earlier, but also to raise awareness of these techniques with the application potential and build these feeder facilities, exploring the frontiers together. And here's another approach. You may be surprised to hear, but we were able to acquire substantial funding, receive substantial funding from the domain of tourism of all places. I came up with a project where we were able to do the science and then leading from there to valorization of this information and a museum exhibition in the National Museum. So sometimes we need to think out of the box to address these key archaeological questions. Here we're looking at broad spectrum revolution, aquatic resources being started to be used, as well as very interesting case studies that can give high profile attention, such as this case study here. And with this, I'd like to close this part and looking forward to your questions. Synchrotron radiation enables us to gather data to provide better answers to long-standing critical questions in human bioarchaeology as well as in many other domains, and critically ask entirely new questions. And I thank you for your attention.
Thank you. Very nice talk. Are there any questions for the speaker? I think we have time for one, maybe two. She's got a hard stop time. So if you want to ask a quick question now, go ahead.
And you can always contact me on the email there. If you're interested in metal exposure or any other topic, you can ask your questions as well.
Okay, well, there's no questions yet. So I tell you what, one will inevitably come to the chat and I'll make sure it gets to you.
Wonderful.
But I know you have a hard stop, so we'll let you go.
Apologies, need to drive for the next one. Thank you very much indeed for having me. And I look forward to helping any of you in your synchrotron journey if you'd so wish. So
okay.
Take care and thanks again.
All right, thank you.
Thanks, bye-bye.
Bye-bye. I know our next speaker is here, Professor Fromm. Professor, you can share your slides.
Yes.
And I think you have a hard stop in 1/2 hour as well, too, right?
No, because it started half an hour. Started an hour earlier than I had anticipated. It's
wait a
5.
minute. Am I doing things out of order? Well, let me check because I could be totally messing up here.
Can you hear me?
Yes. Is your talk for now or 1/2 hour from now?
No, it's from now.
Okay. All right. Okay. All right. So we're good.
So,
if you could, yeah, go ahead and share your
can
screen.
you see me and hear me?
Yes, go ahead. You can start and we can.
I have plenty of time because it's 5:30 a.m. here. Okay, so I will tell you about molecular movies of biomolecules in action with X-ray free electron lasers and status on building the world's first compact X-ray. So I'm from Arizona State University, and in the background you see a picture of our CX drawing. So I'm very happy to give you a perspective of X-ray free electron lasers, which are an amazing instrument. But first, before I want to go to the X-ray free electron laser research, I just want to tell you a little bit about my center. I'm leading a center with 18 faculty in Arizona State University in the Biodesign Institute, and we aim to develop new revolutionary techniques in physical chemistry and biology that reveal structure and dynamics of molecules, with the idea that we want to make visionary discoveries in medicine and energy conversion. And I want to show you in my next slide the faculty. So we have in the faculty a huge variation of faculty. We have more than 100 people in my center. From accelerator physics, this William Graves, to essentially sample delivery technology, to essentially a lot of bio applications from virology, immunology to photosynthesis. But we also combine X-ray studies with other techniques like microelectron diffraction, cryo-EM, and computational studies. I think we have none of the other 17 centers in the Biodesign Institute has such a broad variety of faculty, and we work all together to make visionary discoveries. And I wanted to actually start with a story from photosynthesis. Why we are so interested in photosynthesis? When you would think of, you would be an alien and come with an alien space shuttle visiting the Earth, then photosynthesis is the only process which is visible from the sky. And you see that it's not only happening on land, but also in the ocean, and all the oxygen which we breathe and all the energy which we use, all the food which we eat comes from this process. So we want to know how it works, because when we can build this, when we understand how the natural reaction, for example, of water splitting works, then we could build systems in the future which are as efficient as nature, but as stable as man-made systems. And one of the big protein complexes which makes all the oxygen in the atmosphere is photosystem 2, and this is one of our major focuses of our research. So the challenge is that we have these photosystems, and this is an example of photosystem 1, are really big complexes, and they consist, for example, PS1 consists of 36 proteins and 381 cofactors. So this is a simplified picture of PS1. And the challenge was that it's a large membrane protein, so it took us with conventional X-ray crystallography and synchrotrons 13 years to solve a near atomic resolution structure of PS1. And so we wanted to develop new techniques to determining atomic resolution structures of large macromolecules with nanocrystals. And now comes the free electron lasers into the game. AfLS in Stanford was the first free electron laser which was built in the world. It started operation in 2009, and we did the very first experiments on this free electron laser. I want to tell you a little bit more what is the difference between free electron lasers and synchrotrons. Free electron lasers are huge machines. Each of them has cost more than $1 billion, but they make X-ray pulses that are 10 to the order of 12 stronger than the strongest synchrotron. So what they actually do is they're so strong that they destroy any solid material. And here in the bottom, you see a paper from Neutzer and Heido published in Nature in 2000, where they simulated how a biomolecule would explode in such an X-ray beam. And you see here the scale is femtosecond. So what happens when an X-ray beam hits a sample is that it rips off the inner electrons first, the S and P electrons, and thereby the rest of the atom is positively charged, and the all these positively charged atoms repel each other, and it happened a process called Coulomb explosion. And so this Coulomb explosion was simulated to take about 10 to 20 femtosecond. In reality, we found now that it takes about 30 femtosecond because water dampens the explosion. And so when you compare such an X-ray beam to the laser's worth of Darth Vader, then this Darth Vader's worth is peanuts against an X-ray beam. So it destroys any solid material. But our idea was that we would get pictures and diffraction patterns of biomolecules before the X-ray beam would destroy the sample. And because you outrun the damage, you can do the experiments at room temperature. So we have tried to get funding for 10 years from 2000 until we did the experiment, and we didn't get funding because they all said this is impossible. They don't believe that you get a picture of the molecule before it is destroyed. They don't believe that we could evaluate the data, even if we would see something. And they also thought that it would be really difficult to see diffraction because we wanted to use nanocrystals which contain only thousands of molecules instead of trillions of molecules we need at the synchrotron. And then we did the experiment anyway, and we did it with photosystem 1, this large membrane protein complex I just showed you in the last picture. And to give you an idea, this femtosecond is 10 to the minus 15 seconds. And so the time difference between a femtosecond and a second is the same as between a second and 32 million years. So we did the experiment, the first experiment with photosystem 1. We got millions of diffraction patterns. So you see here the jet. So you have to replenish the sample between each shot. And what you see here, this glow, this is actually from the plasma formed when you hit the sample with the X-ray beam. So the temperature is higher than the inside of the sun. But then what you see here on the side is actually a beautiful diffraction pattern of PS1, and we have even saturated the detector with these small crystals. And this is a back detector, which was here in the middle. And Zeidos have proposed in 1952, when you would ever diffract a crystal where you can count the number of building blocks, that means the number of unit cells, you would see fringes between the peaks, and you actually see this here. And there are the shape transform of the size and the shape of the object, and we just evaluated one of them. And you see that this was a 620 nanometer crystal, so small that you could not even see it under the microscope. And because the wavelength of this first experiment was 8 angstrom, so we solved a really nice structure of PS1 as at 8 angstrom. And so we then developed injectors for the sample delivery at room temperature. We developed methods to characterizing these nanocrystals because you cannot see them under the microscope. Then also new tools for data evaluation, and phasing will become possible with shape transforms. So then we did also the next experiment on photosystem 2, which changed life on Earth as we know it by evolving all the oxygen. And let's see if this video works. Yes, this is actually how the diffraction patterns come in. You sometimes hit a crystal and sometimes you don't hit a crystal. But when you hit a crystal, it looks like, like a firework. And then you have to find the orientation of all these images to be then able to get this structure. And in this very first experiment, we excited the crystal with 2 flashes on the way to the X-ray beam. So this works like that. You have the slurry of the small crystals, which you cannot even see under the microscope. Then comes the X-ray beam. And then we triggered the reaction with the green lasers with every second pulse. So this doesn't trigger, this trigger, this doesn't trigger, this trigger. So we get with 120 images per second, we get alternating light and dark images. So we got 60 dark and 60 light images when we hit a crystal with that X-ray beam. So then this was the very first paper we published in 2014 on this, and we saw large conformational changes of the manganese cluster, even at this low resolution, because the data quality was very good. And we proposed that a water molecule would come in here, like proposed by Isoba et al., and this would form here the oxygen-oxygen bond. These are my 2 students working at this time. at the project. So Shibom is now leading a beamline at the synchrotron and ESRF on serial crystallography, and Chris Kupitz is now lead bioscientist at AfLS in Stanford. So practically all my early students now work at synchrotrons or X-FELs. So then our competitors in Berkeley published a really nice paper where they looked at the stages of the oxygen evolution in the process of the oxygen-oxygen bond form. But the problem is they saw only one oxygen disappearing, and forming the H-oxygen-oxygen bond, you need two oxygens. So there are a lot of exciting questions remaining. So how is the oxygen-oxygen bond formed? What conformational changes happen? And also, what are the oxidation states of these manganeses? So is, for example, manganese 5 involved in this transition state? And so we are at the moment investigating exactly this stage and these questions, with giving 3 laser excitations, and then after different time points Tx, we hit the crystals with the X-FEL beam. And this is actually a more complicated setup than the original one, where we have now essentially a X-ray spectrometer here. So here's the spectrometer, here's the detector, and then here the sample comes from here and the X-rays from here. And so we got a really nice electron density of photosystem 2. And we saw also for the first time a shift in the manganese cluster. And here the trick was that we went to really short pulse duration, because also the people from Berkeley have tried to see if manganese 5 can be detected. For 20 years it was proposed, but nobody saw ever a shift to a lower energy, because they use pulse durations of like 40 femtosecond. And the first thing which is damaged in an X-ray beam are actually metals. So that means when you have too long pulse durations, you would oxidize the metals by light, but you would reduce the metals by the X-ray damage. And so when we went to pulse duration less than 15 femtoseconds, and we saw a clear shift of the manganese compared to model compounds where every manganese is manganese 4. So this is the first indication that manganese 5 might be involved in the water splitting process. And we have more beam time coming, where we hopefully can increase further the signal to noise ratio. So then we have also many other examples of time-resolved studies. We published more than 40 papers in Nature, Science, and Cell on static structures with X-FELs, but molecular movies are the terminal goal, because all biomolecules are highly dynamic, but all what you get normally with the cryo-EM or X-ray structure is a static picture. So here you can actually, with the X-FELs in real time, look at reactions. So for example, this was a dancing of an aptamer, that's a ribo switch. And then I want to focus on this enzyme here, because a lot of people said it's great what you do with the photosystems, but most of the systems are not light driven in biology. So what you do there. And so I want to show you here the results of visualizing enzymatic reactions in action. And this was actually a paper published in BMC, but we have also published more papers on that in the meantime, about antibiotic cleavage by the bacterial pathogen protein BLAC. So this is one of the proteins responsible for drug resistance and antibiotic resistance. And so what you do here, you take the crystals in a jet, and then you mix it at this point here with the substrate, in this case, the antibiotics. And then from the time of flight between the mixing and when you hit it with the X-ray, these are your time points for the reaction. And so we get a molecular movie of the reaction on the enzyme. And the interesting thing is, what nobody has proposed before, is that first the antibiotics comes in and bind in an unproductive conformation. So we have a distance between the serine, which later forms a covalent bond, and the drug of 3 angstrom. Then there is, in 50 milliseconds, there is a rearrangement of the molecule, and then in 100 milliseconds, a covalent bond is formed between the drug and the antibiotics, and then cleavage takes happen. Here is a long time steady state picture. So I've shown you now how cool things you can do with a free electron laser, but there is one big challenge, okay? And this is actually the challenge of the cost of the free electron lasers and of the, let me just make a short thing here, taking this off. So one of the challenge is the cost of the free electron lasers and that there are only 5 in the world. So free electron lasers are linear accelerators. And thereby you can in principle do only one experiment at a time. They can do now some multiplexing, giving some of the pulses to one beamline and some of the pulses to the other beamline, but in contrast to synchrotrons, an AfFL is a linear accelerator, and thereby the number of experiments you can do at the same time in the world currently with AfFLs is only 5, because we have only 5 AfFLs. And each of these AfFLs is multiple kilometers long. The longest is actually the European AfFL. It starts in Hamburg and it ends in Schleswig-Holstein, so a different state of Germany. So it's actually 4.5 kilometers long. Sackler is about a kilometer long, PAL AfFL also. So they're huge facilities and extremely expensive. So European AfFL is the first superconducting one. So therefore also huge amounts of helium needed. And LCLS is now building a second one, LCLS2, which just started operating, which is also superconducting. So really big facilities. And so the beam time you can get is limited. And then in 2014, we were frustrated because we were the first people doing these X-ray diffraction experiments and time-observed X-ray diffraction experiments at LCLS, but then it was super difficult to get beam time after we published the first papers because everybody wants to use it. So what does a biochemist does when he didn't get enough beam time at these fancy X-ray free electron lasers? Our idea was pretty crazy. We built our own one, okay? Which is pretty crazy. So you see here the picture LCLS has cost $1 billion. And here's actually our first compact accelerator, which we built at ASU. So in 2014, I went to our President Kroh from ASU and ran this idea by him. That we have in principle with the BioXFL Consortium and Science and Technology Center drives the implementation of X-ray free electron lasers used for biology, but now it's not enough beam time left, and these big AfFLs are really expensive. So the idea is we want to build 2 compact accelerators. In the first stage, the compact X-ray light source, which already has femtosecond pulse duration, and then in the second stage, the compact fully lasing AfFL, so which would increase access to AfFLs for all the people working on bio research, custom design also for bio application, and then also combine it with optical spectroscopy, X-ray spectroscopy. Then the idea is for the second one, we would reach sub-femtosecond pulse duration. That means we overcome any damage limitations like what I've shown you for the manganese cluster. And the lasing version then was designed with 4 end stations. So the first step, we said we will build it in 2 steps, the compact X-ray light source first. And the president asked me how much would it cost, and I said about 10 to 20 million. And so 2 weeks later, I had the offer letter. You get the center. We built a new building, which you actually see in the background of Biodesign C, with space for 2 compact AfFLs. Because I convinced him that when we build the first of its instrument in the world, it will never be perfect. So we have to optimize it, but then we cannot do experiments. So the idea was we first built the first compact X-ray light source with funding from ASU in this new fancy building with space for 2 compact accelerators. And then we will try to get public funding for the second stage. So this is actually the floor plan here with first 2 compact accelerators. So the walls is where the accelerator goes. It's only 10 meters long. So really compact compared to the big ones. Then here is a hatch. It's a hatches side by side so that we can use laser equipment and so on, both hatches, hatch control. Then this is the laser room and then the RF room. So how does a compact AfFL actually works? So this is here the picture of Biodesign C, and this was how it looks in 2019. And then we starting already assembling it in 2020. So it was also a little bit of a challenge because that time was during COVID. So there were only two things exempt from not working at ASU at this COVID acute time. That was when you have to take care of an animal or if you build a compact X-ray light source. So this is how the first one of the two looks like. So you have here the gun driven by essentially a laser. And we started with UV laser, but currently we run it actually with second harmonic generation with green light. So that's actually the first accelerator in the world operated by green light. And green light is so much easier to handle, doesn't damage anything, and we get much higher flux. So it works like three green photons are converted into one UV photon directly at the surface of the electrode. Then we have three very compact linacs driven by these klystron power supplies. So by this distance, which is about 2 meters, we have already relativistic electrons with 35 MeV. And we run the accelerator at 1 kilohertz. That means we have 1,000 pulses per second. Like, for example, Sakla has 30 pulses per second. PAL-XFEL has 60 pulses per second. We have 1,000 pulses per second. Then we have here several magnetic focusing optic things. And then here we have the ICS shimmer, because our accelerator works by replacing these magnets, which over a distance of a kilometer accelerate the electrons by forcing them on a wiggling path. Thereby they emit X-rays. The big X-rays work like that. So they are on a wiggling path, they emit X-rays, and then the electrons interact with their own X-rays, and thereby they are emitting then at the end, when they're all coherent, there is a very short X-FEL pulses. This is how all the big X-FELs work. That is called self-seeding or Sase principle. So ours had a total different thing. What happens here is, in this interaction chamber, we have the electrons, 1,000 per second, with 35 MeV and about 300 femtosecond pulse duration per electron bunch. And then we collide them heads on with an ultra strong laser beam. So this laser has 200 millijoules. And when the two collide, then this practically kicks off the electrons from their flight path, and thereby they emit these ultra short X-rays. And then the electrons go in the beam dump and the X-rays continue into the hutch. So that is the first one, which we have. And then after the success of building the first one, we have then applied for a grant for building the second one. And then we got 2 years ago 19.8 million to get essentially to build the second one. So while we are at the moment commissioning the first one, we are building already the second one. And the second one is also unique because, let me hide the floating panels. Because the second one is a soft X-ray machine. The first one is a hard X-ray machine. So we at the moment operated at 9.3 kV. The second one is a unique attosecond soft X-ray source, so from 250 eV to 2.5 keV. So it will be fully coherent in space in all 3 dimensions. I have to say that the big X-FELs are only coherent in 2 dimensions. The compact X-FEL at ASU will be coherent in all 3 directions. The pulse duration will go into the attosecond regime. We aim for 300 to 500 attosecond. It will have a very high stability, so no pulse-to-pulse duration difference. And we will have unique control of X-rays of phase and amplitude. So we will accomplish making it lasing by electron bunching via emittance exchange. So we diffract first the electrons on a graded silicon crystal, and then we have them coherent in space, and then we use emittance exchange to make them coherent in time. And here you see the intensity of synchrotrons, the intensity of the big X-FELs, and this is how we will fall with our compact X-FEL. So this is the second one, and which we are building currently in the second soft X-ray suite. And so tunable, we will be able to study all these different transition metals. We will have attosecond pulse duration, which is faster than the core transition. We have extreme stability and variable bandwidth. And the extreme stability we have already seen with number one, this is the AfLS. The big X-fields have shot-to-shot variations of the pulses of 300%. We have less than 0.1%. So each pulse has the same intensity. So then we would combine X-ray diffraction studies with these short pulse time resolved crystallography with SAX-VAX, with optical spectroscopy and so on. So what we can do then with these CXFEL and CXLS, our two accelerators. So we can look at the process of photosynthesis, learn from nature how biocatalysis works. But we can also look at materials. So carbonate reservoirs, oil and gas can be investigated. And we have also look at climate change processes and materials which can, for example, fix CO2 and thereby hopefully remove the CO2 crisis of the energy. And the interesting thing of these bioenergy or energy-related thing is that already the first version which we have is fully tunable from 4 kV to 20 kV. And we can also go to 40 kV. So that means at this high energy, you can penetrate even materials like concrete and investigate, in principle, for example, these microstructures which contain oil and gas. And look at these microscopic pores. And knowledge of the structures are key to, for example, recovery of these natural resources like oil and gas. Then we have also a lot of health impact. We can design more effective drugs with far fewer side views with molecular movies, because we would not get only a static picture of the molecule, but a molecular movie of the molecules. And also for semiconductors, it might be really interesting because the latest transistors are too small to analyze by conventional methods. And with the ASU compact X-ray, we can make extreme UV lithography or look at critical dimensions of semiconductors with small angle scattering. So Intel is also really interested in getting a compact X-ray. So we will have at the CXFEL, we will have 4 end stations, 2 AMO, 1 quantum, and 1 bio end station. And so to summarize, we have essentially, this could lead to new paradigms for energy efficient computing, new materials for the topological magnetic storage, sensitive, high-speed, adaptive materials for optoelectronics, efficient light harvesting for clean energy, what I showed you on the photosystems, development of highly efficient catalysts, and development of novel drugs based on the discovery of the dynamics. And with this one, yeah, I wanted to summarize the different applications. We can also look at matter in extreme conditions, because the beamline which we have can be modified for different materials. At the moment, we have a vacuum chamber and a vacuum detector chamber, but it can also be replaced by a chamber which would contain, achieve high temperature and high pressure. And this is here the CXFEL team in our facility, and I want to specifically highlight Bill Graves, our accelerator physicist, Mark Holl, our chief engineer, and also Sam Teitelbaum, which actually is responsible for the so-called overtaking geometry, which we will realize for getting higher flux for the CXFEL. And then also Robert Kindle, which works on the development of user operation, because our facility will transition into a user facility, so everybody can apply for beam time in the future and conduct experiments. And this is my research group at ASU. And I want to acknowledge funding. So we had also a project in Europe, but Access called, but for this one we didn't get X-rays. So Access is at the moment finished. It was funded by the European Research Council. Then we had a lot of funding from National Institute of Health for the first bio experiments. We had for 10 years the NSF Science and Technology Center, BioXFEL. Then we got 4.8 million for designing the compact XFEL, and then 2 years ago, 90 million for the construction of the second one, the lasing one, and also upgrade of the first one. And then we finished the first one with also 10 million donation from Annette and Leo Boyce. So the first one was built by 10 million from ASU and 10 million from Leo and Annette Boyce. And I thank you for your attention.
Thank you. Are there any questions for the speaker? Is there any in the chat? Anything in the chat? So do you have any schools and workshops scheduled for 2026?
At ASU, yes, we actually plan to have a workshop in the beginning of 2026 on the applications of the CXLS and the CXFEL. And yeah, when anybody wants to come, that would be very interesting for you to attend. So the cool thing about the compact XFEL was the project that we want to bring practically high-tech X-ray technology, which can be used for all these applications, which I said, from these national facilities to a lab. And we have a lot of interest also from current synchrotrons, for example, from ALBA, from also Australian synchrotron and the Taiwan synchrotron, to build a small compact XFEL more or less at the site where they have their synchrotron, because you can do these new unique experiments without X-ray damage, all the structures determined at room temperature. And the compact XFEL is significantly cheaper than a synchrotron. And what we have also planned is to, in the future, when we have built the first two, that we will have a non-profit company which will build custom-made compact XFELs for different applications. So, for example, for Intel, you can also use it for biomedical imaging, so major hospitals may have one. And what I thought would be very interesting, you may want to have one in Africa, because a synchrotron is a huge endeavor. A compact XFEL would be affordable. The first one cost 20 million, the second one 90 million, but with 4 different beamlines. So the accelerator itself is only about 20 million. The most expensive part is actually the laser. So the laser which collides with the X-ray with the electron beam will be 15 million for the second one. That's our biggest ticket item. Because it will be even 5 times stronger than the first one, and there we get higher flux. So I think that would be a very interesting thing for your initiative, that you should not only think of synchrotrons, you can probably afford also build a compact XFEL more or less parallel with your synchrotron. Or start with a compact XFEL.
Yeah.
Then build a synchrotron later.
Yeah, those types of conversations have been had for sure.
Yeah.
Ed, I see your hand is up.
Yeah, hello. Thanks. It was a really nice presentation, Petra. I'm rather jealous. I was just wondering if there is any. You sort of answered the question partially. What are the plans to commercialize these beasties?
Yeah, so our idea is we have another practically 3 years for finishing the CXFEL. We have a 5-year plan for that. Then we have, in the meantime, while we are building the second one, we are fully commissioning the applications of the first one.
Right.
And then we are planning to to have a spin-off non-profit company, which we will build very likely at the ASU Research Park, which is south of Tempe and Chandler. Yeah. And then we will be able to hire, when we get startup funds, to hire new people and build compact XFELs for customs applications. So let's say you want to have one for bio, then you don't need the AMO and quantum end stations, okay?
Right, okay.
Then you would get the accelerator itself for about 20 to 30 million, depending if you want to have the lasing or non-lasing version.
Right.
And then the end station is about like 5 million each. And So that means the total cost for that would be maybe 30 to 40 million. And we would build then multiple of these instruments for customs applications. For example, detector metrology will be even cheaper because you don't have to
Yeah, it
focus
gets cheaper. Yeah, sure. You get
Yeah.
simply. Yeah, okay, and you get critical mass with like effectively mass production. But you
Yeah.
said you'll set that up as a not-for-profit, which is quite remarkable for the US. Why?
Yeah, we want to do this. Why we want to do this non-for-profit? We don't know if it really works, and our investors would, as investors, would go with that. But our Finster plan is because we don't want to charge users for the use of it. Otherwise, nobody comes because all the synchrotons are free.
Yeah, okay.
So the idea is that the revenue from selling these compact
Okay,
versions
got it.
of the
Okay, got it.
X-Ray
Yeah.
will go back for paying for maintenance and operation for providing
Your-
our beam to the users. That's our
Okay,
that's our idea.
Okay, so
If
it's a
this would
globally, it's a
work-
not-for-profit. Yeah, that's quite a nice. I like that. Okay, it's cool. Thank you.
So that's our plan. So that the money which we would get would be used for operating and maintaining and upgrading our instruments. Because when we build the second one, which is fully lasing, then we will also upgrade the first one to a fully lasing X-ray.
Okay.
So at the end, we will have maybe in 7-8 years, we will have both the hard X-ray side and the soft X-ray side being fully lasing.
Sounds nice. Thank you.
Okay, are there any other questions for this speaker? Can you make sure you send me the information on the workshop you're having? I guess it's in person at ASU?
We will have it hybrid.
Oh,
So
okay.
we will have it, we have some speakers will come in person and some speakers will come hybrid. You know, the cool thing about having it in person is that you can actually see the instrument, you know, in live. Also the AfLS is already operating. So you could see this compact accelerator, and everybody who visited us was very impressed, saying this is really compact. And also the fact that we build it in the basement of a building where we have 4 stories of research labs on top of it gives you really short distance to go from, for example, the lab where you make your crystals downstairs. We have also downstairs dark labs, crystallization labs, and so on, where we have the instruments. So it is, as I said, it's very compact and it can be built practically at any campus. So it's not like the big ones where, digging the tunnel costs you already 100 or 2 million. So, the accelerator is only 10 meters. The hatch is actually big. The hatch is as big as the big X-Ray. You see, it has been designed by the users. we have a very compact accelerator, and then we have a nice big hatch where we can do the experiments. Yeah, that's actually the plan for the
I'll be in touch,
commercialization.
and allow me to help advertise. So if there's no
I
other
think
questions for, go ahead. I'm sorry.
it would be for you specifically really interesting to visit ASU and see the instrument, because seeing is believing. You know, so.
Yeah.
But everybody else can then also online participate in our workshop.
Yeah. Okay. Well, let's thank the speaker. And if you want to follow up with her, you can put your questions in the chat, and I'll make sure that she gets them. Before we move on to the next speaker, just give another round of applause. Ed, are you still here? Did Ed leave?
Yes.
Did you see my message?
Email or chat?
Chat.
No, hang on. Over there. Can you intro the next speaker?
You don't have to read it.
I don't know who the next speaker is.
So one important thing which I would just want to mention is that we built the first of the accelerators actually with a workforce of mainly undergraduate students. So we have 30 undergraduate students, 4 graduate students, and 3 scientists really working on building it. And when we had the evaluation of the big grant, the referee said, wait a minute, you built this accelerator with undergraduates? You mean PhD level scientists? We said no, undergraduates. And we recruited them already in their freshman year. So it means that we are also training the workforce. And so when you would want to build one, we would be happy for your students to come to us and be trained in everything which is needed to build an accelerator, which also reduce the cost dramatically because, PhD level scientists in a national lab cost a fortune compared to an undergraduate student in a public research university. So, and the students were really good. They worked really well, and many of them are now went on to their PhD or stayed with the project as staff scientists.
That's brilliant. Thank you very much. I think Lawrence has just dipped out, which is a surprise to me. So it's my pleasure to say thank you to Petra. If there's no further questions, we'll move on. And next up, we have Dr. Marine Cott from the European Synchrotron in Grenoble, who is our beamline responsible for beamline ID21, which is microspectroscopy analysis. And she's got a huge experience in using synchrotron radiation for cultural heritage, ancient materials analysis, very, very fine analysis. And she's also the all-new group head for, what's the name of your group these days? Spectroscopy. Has a special name, right?
X-ray spectroscopy.
It's just X-ray spectroscopy. And she's going to talk to us now for the coming session on looking at how artworks can be studied using synchrotron light. All yours, Marine. Thank you.
Thank you, Ed. Good afternoon, everyone. So indeed, my talk will be a bit, we'll have some overlap with Kirsi's presentation, but in my talk, I will focus more on cultural heritage, not natural heritage, and so meaning manufactured materials. So this is what this is what I plan to cover during this talk. I will start with an illustrative example showing you how micro X-ray absorption spectroscopy can help us getting information about quality of pigments used by Vermeer in The Girl with the Pearl Earring. Then I will switch to Leonardo's paintings and how X-ray diffraction technique can be used to get information about painting materials. And I will conclude with some general words about the SRF and how we try to make the access to ESRF easier for the study of cultural heritage materials. So let's start with a first example, the case with The Girl with the Pearl Earring. So this, what I'm going to show you, is part of a very, very large study which was launched by the Museum Mauritshuis in the Netherlands. And here you have the picture of Abby van die Verre in front of the painting. And here I wanted to illustrate the typical questions that conservators and curators they have when they launch research projects on artworks. On the left, you have typical questions about how the artwork was produced, which were the materials which were used and what were the processes, the procedure, and so on. On the right, you have more questions about how the artwork evolved after the creation itself. Sometimes there are some intentional evolution, like when you have oil drying, but sometimes you have also degradation, or you have some restoration campaigns which may have improved or impacted negatively the artwork. So these are the kind of questions that we try to tackle. So for this research project, the museum installed, you see here, a glass house around of the painting, because I wanted to keep the painting visible while the scientists were running some experiments. So you have researchers who came from all over the world with portable instruments to scan the paintings using different techniques to reveal different, let's say, chemical contrast and information. And here you have a patchwork of the different results obtained by the combination of these techniques. And this is really a direction in which many colleagues are going to develop and use portable instruments for in situ characterization. But at a certain point, this approach has some limits, and occasionally we can have access to fragments to get better, more precise chemical information about the composition of the paintings. So on this image on the left, you see a couple of points which corresponds to locations where fragments have been sampled over the years, so through three different sampling campaigns. And on the right, you see a list of techniques which have been used to characterize these fragments. So most of them are laboratory-based techniques, but 2 of them are synchrotron-based techniques. For those of you who are not familiar with sampling artworks, here you have a movie which sketches the principal idea is that typically when we are allowed or when curators take fragments from the analysis, they are typically embedded into resin and polished transversal to the surface such that you can see the stratigraphy of the sample, and such that with using microbeam or nanometric beam, we can analyze selectively each of the regions in each of the layers of the fragments. So more particularly in this case, we were interested in the blue used by Vermeer to create the girl. And one of the blue used is named ultramarine. Ultramarine, this is a natural pigment. And here you have a sketch of the way it is produced. It is extracted from a rock, which is lapis lazuli. And here you have the sketch of the way the pigment is extracted from the rock using different organic binders and grinding and so on, and with different grades depending on if this is the first, second, or third extraction. What is interesting to note is that if the rock is preheated before extracting the pigment, you will have a different color. The blue is a bit darker blue, while if you don't heat the rock, that's, let's say, a medium blue. And so one of the questions was, can we know which kind of quality Vermeer used in the paintings? And can we determine the quality, and are we able to say if the rock has been preheated or not? And somehow behind that is determining the quality of the pigment. So to answer this particular question, we used synchrotron-based micro X-ray absorption near-edge spectroscopy at the sulfur K edge. Indeed, the main species responsible for the blue color are made of sulfur. And more particularly here, so you see a collection of xanes of sulfur-based compounds. Here sulfides, so reduced sulfur, here sulfates, and this one here corresponds to lapis lazuli. So lapis lazuli is based of a mixture of both reduced sulfur and sulfates. And more particularly, you see this complex feature here can be decomposed into different peaks, and these peaks can be attributed to different sulfur-based species. And according to this to this paper here, the blue color is due to S3-sulfur radicals. So what is interesting to note, and this was a work carried out by Alessa Gambardella, is that when you heat the lapis lazuli rock, you will modify sulfur speciation. And this is what you see on this spectra. On this zoom here, you see the peak intensity is different if you are analyzing a rock which has not been preheated or a rock which has been heated at 600 to 750 degrees. So having this information, we wanted to see if we can get similar signature in fragments from the girl with the pearl earring. So here you have the location of the 2 samples which were analyzed, and on the left part, an optical microscopy image of the cross-section. So indeed, it's not a pure blue layer, it's a superimposition of white, black, and on top of the black, a blue layer. And even in this blue layer, you see that the sample is very, very heterogeneous, and it's very important to be very careful of the location of the xanes analysis. So typically, the way we run experiment is that we are going to define a region over the layer of interest, and we are going to collect a micro X-ray fluorescence map that you can see here. And ultramarine, the lapis lazuli pigment is made of sulfur associated with silicon and aluminum. So if you show the red, green, blue superimposed image of silicon, aluminum, sulfur, the white regions will correspond to regions where we expect to find lapis lazuli. And so after having collecting this fluorescence map, we have selected points, which are all these crosses that you can see here, and we have acquired xanes. And in this case, we found occasionally in some region a signal similar to the one acquired on a model ultramarine pigment, which has been preheated. So our conclusion in this case was the fact that to paint the girl with the pearl earring, Vermeer was using, let's say, a high quality or more expensive pigment, darker, obtained by heating up the lapis lazuli rock. Here, it's just to tell you that we have many cases where X-ray absorption spectroscopy is used at ID21, the beamline for which I am responsible, and for the reason that we cover a very large range of elements, in particular 3D transition metals. So, and sulfur, as in the case of the lapis lazuli. So on top, you have many cases where the question is related to degradation of pigments, change of color associated to change of chemistry. And in the bottom, many cases associated to the study of, let's say, glasses or ceramics, where the question here is to see how the firing procedures will modify the chemistry of the glass or ceramics and consequently the optical aspects of these artworks. So I was mentioning the fact that we can analyze both questions of how an object was manufactured, but also how it degrades. So very briefly, an example about what X-ray absorption spectroscopy can tell us about material degradation. This is in the case of the work of Letizia Monico on the degradation of chrome yellows, in particular in the sunflower paintings by Van Gogh. So here again, they made an extensive characterization at the scale of the painting. And so, for example, in this case, you see fluorescence map of lead, chromium, zinc, and this was used to locate the places where Van Gogh used what we call chrome yellow, which is lead chromate pigments. But in addition to that, some samples were taken, and this is what is shown in this arrow, prepared as cross sections again, and we wanted to see if there was any sign of chemical degradation on this fragment. So again, we started with fluorescence maps to locate the elements of interest, sulfur, chromium, and lead. And from that, we perform some X-ray absorption spectroscopy, in this case, at the chromium edge, K edge. And what you see on this graph is that depending on the location of the point where xanth was acquired, the signal is different. In particular, you see that in some cases, this pre-edge peak is almost absent, as in the case on this point here. And we attributed this evolution to the reduction of chromium-6 into chromium-3 and somehow to the degradation. This case is particularly helpful in the sense that if you set the energy of X-rays at this energy, you will excite predominantly oxidized chromium. While if you excite the same, let's say, if you run the same X-ray fluorescence map, but at this energy here above the edge, you will excite all kind of chromium. And if you compare these two maps, then you can get an idea of the distribution of reduced and oxidized chromium. This is what we did. And very interestingly, you see that we observe at the surface of the yellow layer, so meaning at the surface of the painting, a very, very thin, degraded, reduced chromium layer. So this is something that is very illustrative of the type of experiment that we run on material degradation and notably pigment degradation. I will switch now to a second type of experiments, this time using X-ray diffraction techniques, and in this case with the study of Mona Lisa. I will make it very short, but just to give you an idea about why and what we found during these experiments. There was a unique sample which has been taken in the ground layer on this part of Mona Lisa panel, so on a part which is hidden by the frame when it is presented in the Louvre Museum. The idea of analyzing this fragment was to know more about how Leonardo prepared his panel before painting Mona Lisa. And so we wanted to get information about the composition of what we call the ground layer, which is a white layer applied on the support before the painting. We found in this ground layer materials which are very, very standard and which are the main components of the lead white pigment. But we also found by X-ray diffraction analysis, so high angular resolution X-ray diffraction performed at ID22, we found in some regions here or there very, very, very weak peaks that we could attribute to a compound which is quite unusual, which is named plombonacrite, which is a lead carbonate oxide hydroxide. And this was very surprising because before that, these compounds have been detected maybe 2 or 3 times in other paintings. It's not something extremely common. And so we were searching for explanation of the presence of this compound, if it was intentional that Leonardo used it, if he introduces it as such, or if it was somehow a secondary product of the use of something else. And our hypothesis was the fact that it was there in Mona Lisa because maybe Leonardo may have used a dryer, an oil dryer, which is lead oxide. So our hypothesis was that plombonacrite was not here as such, but resulting from the mixing of oil and lead oxide. And we were very lucky that we also got access to a couple of fragments from the Last Supper, also another painting by Leonardo da Vinci, but this time a mural painting, which is in Milano. And so we analyzed also using diffraction, but in this case, micro X-ray diffraction. So the principle of this technique is shown here. In this case, you are performing X-ray diffraction analysis, but with a beam which is, in this case, the size of 2 microns, and you are going to repeat X-ray diffraction analysis every pixel of a 2D map. And so ultimately, not only you know the composition, the nature of the crystallized phases present in the sample, but also how they are distributed. So composition and distribution. And in this case, we were extremely happy and surprised to see that we could detect lead oxide. So we got the confirmation about of the hypothesis we had about Mona Lisa, that Leonardo was occasionally using lead oxide, probably as a dryer. And in this case, we also find something which was detected for the first time in a historical painting, which is shannonite, a lead oxide carbonates. So with this example, again, how synchrotron-based technique can give us information a posteriori about the way artists were producing their artworks. Now, a few words about the ESRF and how we try to improve experiments beyond just data collection. So you may have already heard about the ESRF upgrade, that there were 2 phases. First phase about the construction of new beamline, second phase about installation of a new machine, which gives a beam much more brilliant and with much lower divergence in horizontal. And so it means that basically, thanks to the new source and the improvement of our instruments, we have data collection which is much more efficient. So we can go much faster. In some cases, we can work with a smaller beam and with improved sensitivity. I will say 2 words very specifically about 2 beamlines which are more often used, let's say, which are regularly welcoming cultural heritage analysis, ID21 and BM18. ID21, I already mentioned the works we are doing on pigments. So the girl with the pearl earring was analyzed, or the fragments were analyzed at ID21, the chrome yellow story as well. And here you have the list of the main challenges that we face when we analyze fragments and how we try to face and to solve these challenges. First is that, for example, in the first of chrome yellow degradation, you can have many factors responsible for the degradation. It can be humidity, temperature, pollution, composition, and so on. So the larger the corpus, the better for you to interpret the factors responsible for degradation. Second, as shown here, you see here the green layer is extremely small. In this case, it's like alpha micron. So if you manage to have a beam smaller, you are able to go deeper into the detail of this kind of phenomena and interfaces. So this is why with the upgrade of the beamline, we designed a nanoscope which gives a beam of 100 nanometers, much smaller than what we had before. Once your beam is smaller, it's still important to keep the field of view. So then comes the question about the speed and the sensitivity. And we have also improved our detection system to go faster and faster. Related to speed as well, here, this map, this is like a black and white image. This is a superposition of 2 maps acquired at 2 energies. But in such an analysis, you make the hypothesis that you have just a mixture which is made of 2 components, A and B. But in many cases, the chemistry is much more complex. So if instead of the acquisition of 2 energies, we can acquire maybe 50, maybe 100 energies or 100 maps, then the knowledge we have about the complexity of the system is much higher. So this is also why it's important to collect data faster and faster. Combination of techniques is also important, with the idea that different techniques can bring different insight into the sample composition. And finally, friendliness is also very important. This is why we have developed user-friendly graphical interfaces and workflow for data processing. A few words about BM18 as well. This beamline has been designed for hierarchical phase contrast tomography. It's, let's say, the evolution of the former ID18 beamline, which has been extensively used in the field of natural heritage for the study of fossils. And it has been decided to build a new beamline specifically for the study of large and heavy samples, objects. So as you can see, large means up to 2 meters, heavy up to 300 kilograms, which means that you can come with very, very heavy and big samples. Here on the video, you see a recent analysis of violins. In this case, the question was, are we able to determine the state, the conservation state of some very important historical violins to see if it is still possible to use them during some concerts. And so that's the main advantage of this beamline, is that you can really go from the object side to the, let's say, micrometric scale and get the full picture about the state, internal state of artworks. So a few words about this beamline and what is the strength, or what are the strengths of this beamline. The sample stage is incredible because it has been designed to mount these very big and heavy samples. Also, the experimental edge is very, very long, which allows long propagation distances. And the detector stage has been designed such to open an easy switch from one low resolution or medium resolution to higher resolution, and to make what they call hierarchical imaging. In the last 5 minutes that I may have, I would like to talk about how, beyond just the data collection, we can also work on improving access to the SRF and also improving the outcome or the impact of the data collected at the SRF. So with respect to access, the standard access is shown here. If you want to get beam time, so you are going to write a 2-page proposal, which will be reviewed, technical feasibility, scientific excellence, and so on. And only sometimes one proposal over 2, sometimes 1 over 3 are accepted. And if successful, you have beam time about 6 months to, typically 6 months to 1 year after the submission. And then you bring your samples at the beamline and you get your data. So if I come back to the experiments I presented about Leonardo's paintings, just to let you know, one map ID13 can be obtained in, let's say, 20-30 minutes. One high-resolution X-ray diffraction pattern at ID22 takes about 1 hour. So because of this increased speed, which is typically maybe 10 times faster than what it was before EBS, we started to think that maybe the standard access to the beam time was not the most efficient for this kind of fast measurements. So we were thinking that instead of having, let's say, 5 or 6 groups competing to use the same setup, but only for a few hours, we could think about what we call block allocation groups, access bag, similarly to what the protein crystallography community is now regularly doing. And so we proposed to other communities to use this bag access, and the cultural heritage community was very, very enthusiastic in testing that. And so in 2020, we submitted the first heritage bag proposal with this group of collaborators. So you see, we have people from different countries in Europe which are collaborating, and they share beam time. For access to 2 beamlines, ID22, ID13, very quickly, what do we offer here? This is a high angular resolution X-ray diffraction beamline. So this is where the fragment from Mona Lisa was analyzed. And the main idea with this beamline is that you can get a very precise and sensitive detection of crystalline phases, their identification, and also information about microstructural and structural properties. And so we have 3 days every semester at this beamline. And the second beamline for diffraction is ID13 for micro diffraction. So this is what was shown in the example of the Last Supper. In this case, the important parameter is the beam size, which is 2 microns, and which allows us performing or acquiring 2D maps, XRD maps over thin samples. Why the bag is so successful, one of the main reasons is shown here is that while before EBS, we were happy if we would scan, let's say, 30 samples over 3-4 days, by improving all the steps at the beamlines, like sample mounting, data collection, and so on, we typically analyze from 150 to 250 samples per experiment. And so this is why we can give access to so many users together during the same bag experiment. So indeed, you have other interesting numbers here, number of users per experiment, and on the right, cumulative number of new users on site over time. And you see that in average, we have about 10 users per experiment, which is really, really something great. Half of them usually first-time users and sometimes also remote users. This is also something we try to balance between on-site and remote users. And they are coming from all over Europe and some of them from US or Indonesia as well. We are also organizing different training sessions on-site or online to make sure that these new users, they are trained to process their data, analyze their data. And my last slide is about the impact of this data. And we want to make sure that the data is not going to stay on disk storage. And when the PhD thesis is over, nobody knows that this data exists. So we got money from the Oscars European Call to build a software to collect not only data, but all the metadata related to the experiment. So information about the instruments, information about the samples, and then we want all this information, data, metadata, to be accessible to anyone. So we are building also an interface so that anyone can see which fragments from Leonardo or Picasso or Vermeer's paintings or whatever have been scanned during this bag beam time. And with this, I thank you for your attention, and I will be very happy to answer any question. And of course, to welcome you, if some of you are interested in joining our bag network, please contact us, and we will be very happy to include you in the network. Thank you.
Thank you.
And are you here?
Let's thank the speaker for a very nice presentation. And if you have any questions for her, you can just unmute yourself and ask away. Let's see. Do I see any hands up? I have sort of more of I don't know if it's necessarily a scientific question, but regarding paintings and the lead compounds that Leonardo da Vinci might have used.
Yeah.
Has anyone ever proposed to exhume his body and see what's in his remains, or is that just a taboo subject? He's buried there in France.
I have no idea about that. To what extent the analysis of his body would give information about his painting, to my opinion, will be extremely limited. Because even if you detect lead in his body, there could be many reasons for lead to be present in his body, not only the material he was using, but anything about in his environment. So let me. I know that some colleagues, they are expert in analyzing, let's say, human remains like hair or bones of famous people, in particular to get insight into possible poisoning actions or medical treatment and so on. This is not something I heard about related to artists.
Yeah, they did it for a U.S. president about 30 years ago, 35 years ago. There were a lot of questions about whether or not he was poisoned. But whatever methods they used 35 years ago might be interesting to do it again.
Yeah, we did it in a French, let's say, woman who was very, very close to the king, Agnès Sorel, and we detected mercury on hairs. but it was super difficult to say if this was due to poisoning or not, because mercury at that time was used in many medical treatments. So reaching a tangible conclusion in such cases is not always easy, because one metal can have different roots or origin, and it, yeah, sometimes brings more questions than answers.
Yeah. And so when you analyze a piece of art, they have to somehow break off a fragment. You can't, like, so if you wanted to do the Mona Lisa, you can't take it down from the wall, bring it to you.
Exact. The Mona Lisa will never leave from the museum. Occasionally we have artwork. So as I shown in the case of the violin, occasionally we have objects coming. There are synchrotron facilities where indeed they are a bit, they facilitate the transport and the analysis of objects. For example, in Soleil, they have a dedicated laboratory and storage area for that. For what I am concerned, since I am in charge of a microscope, most of the time I work on fragments, because we have to deal with samples smaller than 2 centimeters. But indeed, in some cases, if you want to get a full picture, you need to analyze the entire artwork. So that's very complementary to analyze the object or the fragments.
I see. Okay. Thank you.
Welcome.
Any other questions for the speaker? I guess seeing none, let me just ask one last question. Are you, is your beamline scheduled to have any type of school or workshop in 2026?
Indirectly. Indirectly into 2026. So every year at the SRF, there is a users meeting, which is in February. Let me check exactly. I think it's 2nd, 3rd, and 4th of February. And during this workshop, so on the first day, Monday the 2nd, there are some tutorials. And indeed, one of the tutorials will be about PyMCA, which is a software that all our users are using to fit X-ray fluorescence data. The 2nd day, 3rd of February, this is a plenary session. So we have the directors reporting about recent developments, and we have keynote speakers presenting different scientific applications. And on the 4th day, on the 3rd day, so 4th of February, Wednesday, there are 4 micro symposia. And we are involved in one of them, which is about neuroscience and neurodegenerative diseases, and how different techniques can be used in this domain, such as fluorescence, tomography, and so on. And the 3 other micro symposia, I don't remember exactly, but I think there is one about from the hub battery, so related to different works in the field of battery.
Okay. Thank you.
Welcome.
Any last questions for the speaker? So we are scheduled now to do a relatively long break, and we reconvene at, let me make sure I don't give you the wrong information. We reconvene at 17:15, that's 5:15 p.m. With a one lecture from the Synchrotron Earth and Environmental Sciences program in the United States, and then 2 talks from Max 4. So we'll see you at 15:15. Thank you very much.
Thank you.
Ed, are you still here?
Yes.
Oh, I was going to say thanks for
That's all right.
for me.
That was easy. I was just a bit surprised.
Yeah.
That's okay. It was a pleasure to introduce Maureen briefly as well.
Okay.
She does a very, very good talk.
Yeah, so this is interesting. So the users meeting will be, it's hybrid as well? The ESRF user team?
It depends. That's a very good question, and I don't know the answer to that. I think yeah, I don't know actually, because we like to encourage people to be on site because that's really where the value is in the exchanges around coffee, the posters, the cocktail in the evening, this kind of stuff, where you get to renew relationship with the users. They see more about what we're doing. So I don't know. I think typically the plenary, which is like half a day, I think, would probably be in dual mode. The stuff coming from the auditorium, but around the user meeting, we have the user dedicated micro symposia and
Right.
training sessions and all this kind of stuff, which is if I'm so bold, because I don't know, nobody else is listening, right? Then
Everyone's listening.
no, then I think they're the most valuable part of the user meeting. And the plenary is great because you've got the ta-da moments and the high level speakers and stuff like that. But the hard value is really in those little micro symposia where you're going into details on in a specific topic or some training or even some hands-on training or training in some software so you can analyze data. It's around the user meeting. So sort of sometimes it's all good, right? Don't get me wrong, but I think the value is in the stuff around it where which is definitely not in Zoom mode.
Well, let me recommend that you invite Phil Majara and Ambassador Ngari from Kenya, the two scientists that are UNESCO, the African.
Yeah, well, I think we've got
scientists.
UNESCO. You got the UNESCO thing coming up in Paris. I don't know who's going to be there. There's a
Yeah.
visit to Soleil that's organized the day before,
Right.
which I'm tempted. I can't get to it because I'm coming back from Hamburg, and I'm not going to spend the week away from family. So I
Right.
go up to Paris on the Thursday, so I'm at least to be sure there for the UNESCO meeting. But they'll see a synchrotron on Thursday. It would be nice if we get a visit to the ISRAP. I mean, that's
Yeah. Send them an invite. They might get on.
Well, let's see how the December visit goes.
Yeah.
We've got the Sesame Council meeting is being hosted at the ISRAP in May next year. So
Yeah,
that may also provide opportunities to add 1 and 1 get 3 or something. I don't know.
Yeah. Okay.
Let's see.
All
So
right.
it depends on, we keep saying we've got time, but then actually in some ways we don't have a lot of time if one wants to push this stuff along. I love the stuff from Petra. That was very cool.
Yeah.
Very cool.
Life comes at you fast when you say you think you have time.
Yeah. Little baby XFEL. That's very cool.
Oh. So I haven't heard actually from the UK XFL people. Like, did they?
I think it's still I'm out of touch. I think there's been a recent, I don't know, but I think there's been some kind of town hall meeting, which is UK or American speak for the community getting together, right?
Yeah.
And I think the UK XFL is maybe still lurking there. I don't know whether they were going for a low or a high energy one, because the UK is a partner in EU XFL anyway. So they probably complement that with a lower energy machine. There's space on the Harwell campus. I guess that's where it would go. I don't know whether the UK has burnt its research funding in doing the Diamond 2 upgrade, which is underway now. Or at least they're doing the procurement getting ready, and then Diamond shuts down for its dark period in something like end of 27 for a short period. So maybe they need to wait till Diamond 2 is done. I have no idea. Don't know, right?
That might be. I mean, a lot of it is you have to sort of make your case to the Treasury.
Yeah.
Treasury might be risk averse at the moment.
Well, it is sort of, there's risk averse and there's risk averse. I mean, it's always a difficult calculation. I mean, do you fund the teachers? Do you fund the hospitals? I don't care about a bit of extra motorway or an airport personally. Or do you chuck a couple of billion at an XFEL? A couple of billion on the scale of the government is, as we've often discussed, is not huge, but you've still got to do strategify, right? And that investment will generate jobs. It will generate technology. And I guess I don't know what that looks like in terms of you chuck $1 billion at science infrastructure, what the comeback is for national infrastructure, what the comeback is for the country in terms of jobs and purchasing power, because you're going to buy stuff from national companies, maybe. Although for some stuff, there's no choice. You have to go to like China or wherever. Anyway, I don't know what the maths look like that. I guess you need an economist to tell us.
Right. Actually, I remember the science minister of Nigeria telling me that who we really should be talking to is the finance minister.
Yeah. Yeah, you need both. You need for some of the stuff recently we were talking about in terms of science diplomacy. You need it's not, you don't need the science and tech minister. You need the foreign minister, because they're the ones that see the point of the science diplomacy, and then they go and talk to the science minister, saying, "Hey ho, let's go," right?
Yeah.
But you're right. It's the finance ministry, and what is the investment you're pitching it, right? You're doing a 60-second Dragon's Den or Sharks. What is it in America? It's a Shark's Tank, right? It's Shark Tank.
Yeah.
A Shark Tank style pitch, say, for $1 billion, do that, and you're going to get back 10 billion in wealth generation through jobs, technology purchases, and spillovers from the science or something. I don't know, but you need to study. Somebody somewhere has done that, right? For all of the RIs that were ever built. I don't think I've ever seen one though.
All right, I gotta make a run, and right. So what time is it now? It's coming up on 9.
Is it? Bloody
So
hell!
we have an hour and a half break.
Okay, it's 3 o'clock in France.
Yeah.
So it's tea time nearly.
It is. Yeah, I need like major coffee time.
Major coffee
Like
time. Right.
I
Excellent.
didn't think I
I've
was
got.
going to make it this morning.
No, well, I'm not surprised that you started early, right? You were in the sessions at 10 or something, right? Which was God knows what time in the morning over there.
9. Yeah, I had to do the 9 o'clock session.
Yeah. Right. Well, I'm going to drop out between 4 and 5:30. I've got to do an interview here.
Okay.
Yeah. Okay, cool. See you later.
All
Yeah.
right. We'll talk to you later.
All right. Ciao. Bye-bye.
Bye.
Hello. Hello. क्या बोल रहे नोट वो मीटिंग चल रहा बोला तो बोला ये वीडियो नहीं दिखा रहा वीडियो कहीं दिखाया करा देखो वीडियो तो यार वो स्टॉप कर दिया आपने हाँ? कर दिया कौन है डाटा पे कार रखा इसको कार है क्या है एक वीडियो वो आपको स्टॉप किया स्पीकिंग हाँ बोल तो रहा है आवाज यू वीडियो स्टॉप अभी चालू है हो गया चालू हो रहा है अभी चालू हेलो 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 तो वीडियो रिकॉर्डिंग डायरेक्ट इस पर बोलेंगे बात करेंगे अब हाँ?
Hello. Hey, Tess, this is Lawrence.
वीडियो रिकॉर्ड कम तो बोल हो जाएगा आपको वीडियो रोक दिया हाँ तुम्हारे वो बोला तो हो जाएगा आपको हाँ तो
Hello, Lawrence.
Hey, were you able to reach Leonard?
No, I'm going to reach him now.
Okay. All right, I'll let you go.
Do I have the Zoom link? I joined through the program, actually.
Just cut and paste and send it to him.
Okay. Time was the CS director speaking.
At in basically 15 minutes.
11.
It's 10:15.
Our time?
Yes, in 15 minutes.
Hello.
Ugh. Bad link. That's a lot of ace.
Hello, I Dr. Krishna Chaudhary, very happy for your program, quantum community program event. African, South African biophysics program. Very nice, very learned from this program. Thank you. I have been attending about 3 days continuous, 17, 18, 19 November 2025. Thank you for very much. Very good. Thank you. I Dr. Christopher Chaudhary, learning for your program, African Biophysics program, conference, very good. I like very much. Thank you for. Thank you, sir. Very good. Very good. Thank you. Thank you, sir. Thank you. Dhanyawad. Thank you. Thank you. Thank you. Very much more. Thank you. Thanks for your support. Thank you.
Let's see. It is getting to be the hour. Let me make sure. Ah, here we are. Hello, Andrew. This is Lawrence.
Hi, Lawrence. Good to hear you.
Good to hear you. So we'll start in about 5 minutes.
Yep, great. Okay. I did upload my presentation last night, but I assume that for Zoom, you just want me to present it from my local slide deck. Is that
Yes. Let me do that. Make you the co-host.
great? Let me find my slides. Is it okay to go ahead and
Yep, you can go ahead and set your
present?
slides and everything. Yep.
Okay. How's that look? Are you able to see everything?
Yes.
Is it in full screen mode now?
Now it's in full screen. Yes.
Great. I understand it's a fairly long break that people might be returning from.
Yeah, we had to pull the plug on one session.
I see. Yeah.
Those too many pieces were falling off the train.
Understood. Yep. Okay. Well, hopefully they'll get back on the tracks here in a few minutes.
Yeah. We do monthly Zoominars.
Ah.
So we could just do it then.
Yeah.
I don't know if you recognize that Tessima is in the chat in the room.
I'm sorry, say that again. Somebody's in
So
the
you
chat.
see Tessima is here.
Ah, okay. Good.
Recent government retiree, I guess.
Yeah.
Well.
Or as they say someplace, a pensioner. He's a pensioner.
Pensioner, yes. Happy to be here.
test did you find, Leonard?
I dropped him an email and a text.
Okay.
It's Wednesday, so Wednesdays are normally some meetings. So at this
That's
time,
probably super crazy there today.
yeah.
And probably crazier still at my old place, O&R. They got rid of the admiral and put in a not an admiral. Hmm. Maybe they try to squeeze NSF back into O&R like it
No,
was in the beginning.
they're putting NSF at the patent office.
Oh yeah, that's right. Yeah. Oh, that's right, because you guys got to vacate, or the agency
Yes,
has to vacate. Wow,
exactly.
crazy times. Okay, why don't we go ahead and get started with our first speaker for this session? Is Professor Andrew Campbell at the University of Chicago, and he's going to speak to us about the Synchrotron Earth and Environmental Science. I guess it's a consortium. It's a good word, and he'll tell us more in his presentation.
Yeah, thanks very much.
Take it away. Yeah.
Okay, thank you. And thanks for providing me this opportunity to tell you more about SEAS, the SEAS program that stands for Synchrotron Earth and Environmental Science. We're a program funded by the National Science Foundation in the United States to support operations and management of a variety of synchrotron beamlines that focus on Earth and environmental science research across 4 different synchrotron facilities in the US, as shown here. SEAS is hosted at the University of Chicago, where I am, but it's done in right where we carry out our work in partnership with a variety of other universities and synchrotron institutions. Uh-oh. Let me try to get my slides to advance. Yes. My goal here is to sort of walk through the variety of techniques and geoscience applications that our users carry out at the SEAS-supported facilities. Some of those are summarized here. We've got X-ray based applications mainly, although we do have one infrared spectroscopy beamline. We provide users in the geoscience community with access to X-ray diffraction and surface and interface diffraction techniques with a variety of X-ray microprobe techniques, including fluorescence and imaging and near-edge absorption spectroscopy methods. We've got several beamlines providing synchrotron X-ray computed tomography applications in either X-ray absorption or phase contrast tomography. There are coherent scattering techniques available, and I'm thinking here especially of a soft X-ray tachography. I've got an application towards the end of the talk to show you. We support one nuclear resonance scattering beamline, and as I already said, a synchrotron infrared spectroscopy beamline. This wide range of synchrotron-based applications finds useful application to a similarly broad range of Earth science research topics. And I'll give examples of several of these as we walk through the talk. But there are a number of geochemistry or petrology, questions that benefit from synchrotron applications, environmental science and low temperature geochemistry applications. I'll show you a couple of those. Soil science, paleontology, mostly this is tomographic studies of the 3-dimensional internal structure of fossils, but there are some microprobe applications to the paleontology community as well. A really big part of the SEAS community comes from study of the Earth's interior through experimental studies. This is deep Earth mineral physics or rock mechanics studies for near crustal deformation, related to earthquakes and faulting or propagation of fluids, as well as petroleum reservoirs and more. Other sort of on the periphery of Earth science, I guess I'm thinking of, atmospheric aerosol chemistry has applications that are beamlines, as well as planetary science, mainly meteorite studies, or in some cases, the examination of samples returned from, spacecraft. As I mentioned earlier, SEAS operates at 4 different synchrotron facilities in the United States. Those are listed here. The Advanced Photon Source, or APS, at Argonne National Lab in Illinois, which is close to SEAS headquarters here. The Advanced Light Source at Berkeley in California, the National Synchrotron Light Source 2 at Brookhaven National Lab in New York, and the Stanford Synchrotron Radiation Light Source on the Stanford University campus in California. The primary goals of SEAS as an organization are highlighted here. First and foremost, it's our mission to operate and continue to develop state-of-the-art synchrotron beamlines for efficient centralized management of these facilities to produce, productive research environment for Earth and environmental science users. We're funded by National Science Foundation, the EAR division, Earth Science Division's directorate specifically for this. We enable new capabilities and provide new research opportunities by taking advantage of technological development in either improved instrumentations that we invest at the beamlines, or by exploiting, or I guess, working with the synchrotrons as they upgrade their own light source. For example, the Advanced Photon Source is just now coming off of a major upgrade that completed in 2024, and we're investing in improved instrumentation to better take advantage of the much greater resolution and brightness of the Advanced Photon Source at Argonne National Lab. We engage broadly with the geoscience researchers to support their research needs, and it's that interaction that sort of drives our thinking of where to invest in staffing and instrumentation to improve these facilities for community use. And related to that, we try to provide a robust outreach and education program to attract and assist these users and to support the next generation of Earth scientists. A few quick facts. SEAS has been in operation for just over 2 years now as part of a 5-year cooperative agreement. We support 36 staff in whole or in part, including mainly beamline scientists, but also, some administrative and technical support staff. As I said, it's hosted at the University of Chicago, and we've got subawards to help manage some of the beamlines at various universities around the country, including University of California, Santa Cruz, the University of Minnesota, the University of Illinois at Chicago, Stony Brook University in New York, and Stanford University in California. Each of these are responsible for managing one or more of the variety of beamlines that SEAS supports. In total, we support more than 15 beamlines at the 4 different synchrotrons around the country. As I mentioned earlier, we also support outreach and education activities to support our users not only when they're at the beamline, but also afterwards and before. We sponsor workshops to promote new techniques as well as train users in established ones. We collaborate with ongoing programs that are run by the facilities or that are initiated by our user community. We support new user programs. We are, like every website, we are continually improving in our web portal to provide beamline information for users. I've got a link at the end of this presentation to the QR code so you can follow it and see seascience.org. We provide outreach to not only our established user community, but also potential new users through workshops and meetings and conferences and webinars like this one. And we host an annual user meeting. Our first one was just held this year, actually in Chicago, and there's a picture of it in the lower right, showing the Chicago skyline in the background. Okay, SEAS, as I've explained, SEAS provides beamline staff and equipment, and we do this in partnership with the synchrotron facilities to negotiate, leveraged access specifically to promote the Earth and environmental science research communities. So we negotiate these agreements with the Department of Energy management of the synchrotron facilities. We are funded by the National Science Foundation not to obtain this synchrotron beam time for our own use, but to make it available broadly to the geoscience community. So we operate to provide access for the Earth and environmental science community broadly. And as a consequence, we have, several hundreds of users coming to use our facilities from around the world every year. And accordingly, the general user system, a proposal basis, is the basis for access to SEAS-sponsored beam time assignments. I'm going to spend most of the rest of the talk sort of walking through a sample of the technical applications and some earth science results that are obtainable using some of the SEAS sponsored beamlines. I'd like to start off with the in situ rock deformation community. This, we recognize, is a community of researchers, at least in the United States, who are sort of poised to take maybe better advantage of synchrotron facilities than they have been doing over the last, couple of decades. So recognizing this, SEAS has recently made investments to provide expanded beam time access, right, to open up additional beamline facilities for this community to take advantage of, as well as investing in equipment for in situ rock deformation studies. One example of that type of equipment is shown here on the right. This is the Mjolnir triaxial deformation cell. It's a cell that you would use, that researchers can use to examine the deformation of rocks under sort of conditions of the Earth's crust, like up to 500 or 600 megapascals and a few hundred degrees C. So these are sort of conditions exploring, fracturing and faulting and hydrothermal reactions in the Earth's crust. Examples of, this is just one of a whole sort of array of deformation apparatus that users can bring, but this is one that we have reproduced at SEAS facilities, and users have taken advantage of that kind of cell. An example of the type of application that you would obtain with this cell is most of these are tomographic studies, and shown here in the middle of the slide, in the lower part, is a tomographic study showing the increasing degree and distribution of new fractures in a sandstone as it's being stressed from, I don't have the labels here, but roughly 100 megapascals to 200 megapascals of imposed shear stress over the course of this study. So at low stresses, you can just see the beginnings of fractures, and then the fractures, increase in number and organize into, one major fracture system running through the sample as the stress is increased. There's a variety of, rock deformation applications based on this type of study, largely using tomographic applications, and also sometimes coupling it with X-ray diffraction to understand the mineralogical reactions that are taking place, combined with the three-dimensional structure of the deforming rock. As I mentioned, we've invested new support for rock deformation experiments at multiple beamlines, at the Advanced Photon Source, beamline 6PM, at the Advanced Light Source at Berkeley, beamline 832, is newly supported by us with a staff person to support rock deformation experiments. We've also can provide support with our existing beamline staff at a neighboring beamline at the National Synchrotron Light Source 2 in New York. That's the Hex beamline. This is a really high energy, high flux, very large beamline that's really exceptional for this type of study. And also, of course, in our effort to support and grow this in situ rock deformation community, we've provided workshops and co-sponsored meetings, such as the one shown below. Here's an example of some commissioning experiments that were done at the Hex beamline that I mentioned in the previous slide. This is at the NSLS2 in New York. The SEAS team was among the very first users of this beamline as it opened up in October 2023. Even on day one of our measurements, we obtained, the team obtained excellent 3D tomographic results showing, in great detail, failure of a granite sample after deformation in another one of these triaxial deformation cells. As it says here, excellent tomography results within 1 day of the very first beam. crack formation, sample failure, and in a variety of rock samples. And we were also able to probe the sample using X-ray diffraction in situ under the formed rock conditions. So, yeah, so that's a sampling of the tomographic studies for the purposes of investigating deformation of rocks in the Earth's crust. Tomographic studies at seas beamlines enjoy a much wider variety of applications than that. Here's an example of a paleontological application. This is computed tomography of fossils. This is a paper that was published just earlier this year by Yara Haridi and her co-workers. It was published in Nature in May, as shown here. The purpose of this study was to investigate the fossil record of the earliest vertebrates in Earth history. So in this study, they used tomography, X-ray tomography at a SEAS-sponsored beamline at the Advanced Photon Source to examine both ancient fossils Cambrian fossils that were proposed as potentially the earliest or among the earliest vertebrate examples in the fossil record, as well as some more recent fossils from the Ordovician and present-day arthropods for comparison. And here's some examples of the very high resolution 3D tomography that they were able to obtain. And the green imaging here highlights these sensory structures that they focused in on as part of the anatomy of these creatures. These figures in particular show the comparison between the Cambrian fossils here on the left and the modern day arthropod fossils on the right. And they illustrated that the features that were proposed as indicating a vertebrate structure in these 500 million year old fossils are actually much more similar to sort of modern day arthropod sensory organs. So it's likely that those were not in fact vertebrates in the Cambrian. So this study has sort of reset our understanding of the history of vertebrate animal evolution, such that the earliest vertebrates that are really known now are moved forward by tens of millions of years up into the mid-Ordovician period. Sticking on the theme of tomographic studies, we've also, our SEAS users have also used X-ray tomography to investigate the origin of very deep forming diamonds. As many of you may know, diamonds form under a wide variety of conditions in Earth's deep interior. Some of them are thought to have perhaps formed from really deep, like in excess of 600 kilometers depth in the Earth, or various, depths in between. Evidence for that is controversial, and in this study, they developed techniques to sort of systematically study a large number of diamonds in an efficient manner to, first of all, identify inside the diamonds, where are the locations of the minerals that are included as the diamond grew. So what they're looking for here is not the diamond itself, but the included minerals. Once they identify the mineral inclusions through X-ray tomography, then keeping this sample where it is, perform X-ray diffraction at exactly that location to identify what are the minerals that exist. And by looking at the crystal structure of the mineral inclusions, one can compare that to what we know about Earth's deep interior to extract information on the depth of origin of these diamonds. And here's an example showing the existence of a mineral called ferropericlase in an inclusion in this diamond. And that strongly suggests to these authors that this may have formed in the Earth's lower mantle at 660 kilometers depth or greater. Okay, the study of Earth's deep interior more broadly is actually a really important and sort of recurring theme at the geoscience oriented beamlines. The study of mineral physics is a study of the properties and structure and evolution of Earth's deep interior. And this is a community that has relied on synchrotron applications for a long time now, not because it's a very large community, but because they really need synchrotron sources to be able to probe the properties of their specimens when they're being held at these really extreme conditions of high pressure and high temperature that exist deep inside the Earth. We've got the number of beamlines listed here. It looks like 6 for diamond anvil cell applications and 4 beamlines where we support large volume press applications. These are the 2 major experimental techniques that are used for high pressure, high temperature research to understand the properties of Earth's deep interior. There's a wide range of applications that are used in situ conditions while the rocks or minerals are being held at these high pressures and high temperatures. They're summarized here. As you can see, diffraction, imaging, deformation studies, combining the radiography or X-ray diffraction along with acoustic velocity measurements or acoustic emission measurements showing fracturing, other transport properties, as well as other types of spectroscopy through the diamond anvil cell. The pressures and temperatures that can be obtained in these conditions are comparable to those that span the Earth's deep interior. The large volume press experiments cover much of the Earth's upper mantle, and then these diamond anvil cell experiments can achieve several millions of atmospheres of pressure and thousands of degrees of temperature, replicating the range of pressure and temperature conditions that exist even into the Earth's core. Here's some examples of the large volume presses. This relies on, powerful hydraulic presses. As you can see here, pressing on the anvil apparatus that squeezes the sample, which is embedded deep inside these experimental apparatus. And the sample chamber here is a fraction of a millimeter in size. Depending on the particular application and what type of property of the rock or mineral is being studied, there's a wide range of tungsten carbide anvil geometries that are available at SEA's beamlines for the deep earth studies. Examples of the mineral physics research are summarized in the next few slides. Much of what we know by observation of Earth's deep interior comes from seismological studies, where we're making measurements on the surface, and from these, seismologists can infer the distribution of density and acoustic wave velocities as a function of depth inside the Earth, and to a limited degree inside the Moon and Mars as well, where there have also been seismological studies. Accordingly, understanding the equation of state, that is the pressure, density, temperature relationship, as well as the acoustic wave velocities, is a really fundamental part of mineral physics. And that's illustrated here by these measurements of compression wave velocity, shear wave velocity, and density as a function of depth, or as a function of pressure and temperature for this particular sample. Whoa, sorry. All right. Sorry for the noise. Yeah. Okay, sorry for the noise, but anyway, shown here is a video showing a radiography measurement of viscosity of rock melt under conditions of the Earth's lower crust to understand the migration of melt in the Earth's crust that eventually form magma chambers near the surface. And these can be done in situ in multi-anvil press experiments. The very high pressure, high temperature experiments are done using a diamond anvil cell. I've noticed some other diamond anvil cell studies on the program for this conference. I think on Friday, I can't remember. But an example of the diamond anvil cell was shown here. It measures roughly 45 millimeters across. The X-ray beam comes in from the right, and there's a detector in the back. This is a very simple sort of diagram of an X-ray diffraction experiment. It can also be combined with laser heating. Shown here are 2 optical systems carrying high-powered infrared lasers that will heat the sample to thousands of degrees while it's being compressed in the diamond anvil cell to tens of GPa or even hundreds of GPa of pressure. That corresponds to, hundreds of thousands or millions of atmospheres of pressure. And shown here is the, I guess the environment where the users sit while the experiment's being performed. In this example, which appeared in Nature Communications earlier this year, these users investigated the phase change, the phase boundary in the magnesium silicate system in the region of 23 GPa. This corresponds to a major phase transformation that exists in Earth's mantle at a depth of 660 kilometers. This transition has been known for decades. It's one of the most important mineralogical transformations in the deep interior of the Earth. Yet still, with improving, precision and improved technological, capabilities, including at C's beamlines, as shown here, there's still, valuable work to be done and more knowledge to be gained. This study illustrated for the first time that the phase boundary has a measurable and significant curvature, which, as they also modeled, has impactful outcome on the dynamics of the Earth's convecting mantle. I know I'm running a little short on time, so I'm going to go quickly through this final application in which the SEAS users discovered new iron sulfide crystal structures under really extreme conditions of Earth's core, reaching 100 to 200 GPa, that's 1 to 2 millions of atmospheres approximately, and temperatures up to 3,500 Kelvin or something like that. Here's an example of the X-ray diffraction powder pattern at 169 GPa and 2100 Kelvin. Going beyond this and getting more detailed infrastructure on the new phase, a newly discovered phase indicated by the H, indicating a new hexagonal phase seen in the powder pattern here. The powder diffraction didn't allow us to solve the crystal structure precisely, but as you can see, the diffraction pattern is really spotty. So these researchers used a technique called multigrain single crystal X-ray diffraction to sort of simultaneously obtain lattice parameters and refinement of the crystal structure of individual grains in the sample, even though there were many dozens of grains coexisting at once. So it's like single crystal diffraction, but it took a lot of work to figure out which diffraction peaks corresponded with individual grains. That process is shown here, where the yellow ones here, the yellow dots are sort of line up indicating reflections in reciprocal space that all correspond to a single grain. And by extracting that information and performing refinement on just those peaks, the authors were able to resolve the structure of the new hexagonal iron sulfide phase, Fe5S2. Here's the structure. As you can see, it's a large unit cell and fairly complicated structure with a significant degree of positional atomic disorder. But the important point here is that this new iron sulfide phase, Fe5S2, is the accessory sulfide phase that will co-crystallize along with metallic iron in the Earth's metallic core as it slowly cools and solidifies. So it's a newly discovered phase that we know, has importance to the evolution of Earth's innermost core. Finally, I want to show a few applications regarding X-ray spectroscopy, which plays a variety of really important roles in many Earth science applications. This is hard X-ray. This example shown here is spectroscopy of hard X-rays. This is around the absorption edge of copper at nearly 9 keV. This study showed through a series of analyses of both experimental and natural samples the importance of dissolved sulfide in controlling copper speciation in basaltic melts. They illustrated that the valence state of copper doesn't depend only on the redox state of the system broadly, but also on the presence or absence of sulfur. Moving down into the more tender X-ray region, here's a spectroscopy study at approximately 2.5 keV. That's the region where the sulfur K absorption edge is. And in this study, the authors were able to map out the spatial distribution of many different sulfur species in corals, both in the organic and in the inorganic part. And through this information, they're able to infer much information about the growth cycle of corals and how it responds to global climate change. And then moving to even softer X-rays, this is in the region of just a fraction of an EV, right? This is 0.3 keV here. This is a study showing spectroscopy of carbon as well as ptychography on the iron 3+ L edge. So the purpose of this study is to explore the properties of particles obtained at deep sea hydrothermal plumes to understand the speciation of iron and the degree to which it can serve as a nutrient for the phytoplankton in the oceans that makes up a really important part of the global carbon cycle. There's a spectromicroscopy shown in the upper right for around the carbon edge showing speciation of organic and inorganic carbon. And in the lower right, I'd like to point out this is a soft X-ray ptychography image with remarkable spatial resolution. You see that scale bar there is 100 nanometers, and it's imaging the distribution of specifically trivalent iron in this case. So providing some insight into the speciation of iron within these colloidal particles that were collected at the hydrothermal vents. Okay, finally, I'd like to point out that SEAS supports synchrotrons for the general user community, supports synchrotron beamlines for the general user community. All of our beam time is awarded through facility-specific proposal systems. So we don't manage the allocation of beam time. The synchrotron facilities do that, and we are just there to support the staffing and equipment to specifically support earth science-oriented applications. You can learn more about us at cscience.org. If you have specific experiments in mind you'd like to pursue, I encourage you to reach out to our beamline scientists, and you can access us either at this URL link or by following this URL code on your phone if you're fast and click that with the camera. Thanks very much, and I'm just about out of time, so I'd be happy to answer any question that you have. Thank you.
Thank you. That's a very nice talk. Any questions for the speaker? I have just sort of a quick question. It's just organizational. The other light sources in the United States have, I guess, capability that either overlaps or is complementary to the ones that are explicitly included in your consortium.
Yeah, that's right. That's right. I mean, yeah, earth science oriented users can, can and do go anywhere that they're able to obtain beam time. In many applications, the particular experiment they're doing requires a certain experimental setup, like the diamond anvil cell or the high pressure experimentation as I described. But for X-ray microscopy or tomography applications, they can go elsewhere, and they do. But at many of these beamlines, it's sometimes difficult to get access for earth science experiments. I mean, if the review panel is managed by a bunch of chemists or physicists or something, they might not appreciate the profound importance of geoscience research. And what we find is by negotiating these agreements with the facilities to emphasize earth science research at specific beamlines, it better supports the geoscience, synchrotron going community as a whole.
Okay.
Yeah.
And do you have contemplate some kind of a school or workshop in 2026?
Yes, we have. We're constantly either preparing workshops or partnering. The first one on our calendar that I can think of offhand is again about rock deformation experiments, and that's being scheduled for March. And at cscience.org, you can find a link that will allow you to sign up for the SEAS email distribution list, where we announce these types of workshops and meetings. Not only the ones that we sponsor, but also anything that's related, that we think would be of interest to our community. We are happy to, broadcast. And we've got nearly 2,000, like 1,950 scientists on our email distribution list who keep up with the earth science synchrotron applications sponsored by SEAS.
Okay. Any other questions for the speaker? And if not, let's give them a round of applause.
Thanks very much for listening.
We'll be in touch because I think what we want to try to do is have an African School of Synchrotron and Earth and Environmental Sciences. We can do it sort of remotely, and then maybe after 1,168 days, we can think about in person.
Exactly. Oh yeah, let's stay in touch and partner on that. Yeah.
Okay. All right. Thanks for your time.
Thank you.
Our next speaker is from Max 4, And I just realized that the pronunciation could be finest beamline, the finest beams.
That is how we usually pronounce it, yeah.
I've been saying, up until 30 seconds ago, I've been saying finest.
Yes, well, it's the Finnish and Estonian, so
Yeah, okay.
It's also correct, but yes, finest beams.
Finest beams, where they do, amongst other things, gas phase atmospheric science studies. And our speaker is Dr. Axel Eriksson. So take it away.
Indeed. So we are going to change gear now from rocks to air. Let's see if I can make this sharing work. How is that?
Yep, you're coming up.
Okay. You're seeing a PowerPoint?
Yes, I am now. Yep.
Is it being showed in presentation mode?
Not right now. Now it is.
Oh, okay. So things are in order. All right. So the title of the talk was in the program. So I'm actually not from MaxLab. I'm from Lund University, which is hosting institute for MaxLab, because the MaxLab employee that was contacted, Antti, was kind enough to pass this honor to me. So I'm an associate professor at Lund University. I'm not the first Lund University scientist you hear in the conference, but I am the first one who does not have a Nobel Prize. So that's me, Axel Eriksson. I work exclusively with air pollution.
Who do we write to about that?
Huh? Yes, yes. Well, I can maybe come back next year.
Okay.
For 2 years. Yeah. So the title was Finest Atmospheric Science Beamline, which is actually not really true yet, but the Finest does have 2 end stations, whereof one is a gas phase end station, and there we have a sample environment for atmospheric science that I will talk a little bit about. Consistently, I try to put the orchids of the people involved in the slides, and if something is missing, you can always find me afterwards to complement. So as a background, I work with aerosol particles, and I've neglected to put in the obligatory why is aerosol particles important slide, but it is a fact that aerosol particles is the largest environmental cause of death on any continent, including Africa. And it is also the largest source of uncertainty in our understanding of climate. So there's 2 important reasons to try and understand aerosol and the effect that they have in the world. And this effect depends on the physical-chemical properties of these particles. So aerosol particle means anything from nanometer to micrometer scale, which is solid or liquid and suspended in the air. And depending on what and where, it has very different impacts. One category of aerosol, which is heavily researched because it's complicated, is secondary organic aerosols, which means aerosols that form from gases under oxidation in the atmosphere. And for that reason, my dear colleague Matthias Hallqvist, he wrote a paper, it is now 15 years old, about current and emerging issues with secondary organic aerosols. And in this review paper, there is a plot I quite like of instrument usefulness from his perspective. So it's the plot that's here. And you can see on the y-axis the completeness, the fraction of the mass of the particles that we get, and the x-axis the resolution. So what kind of information we get? Do we get molecular ID, or do we get other classes? And then it's colored by other desirable things, to be specific time and size resolution. And then Matthias has indicated that the perfect instrument for him, it gets the mass, 100% of the material is analyzed, and there is molecular information. I don't think that's a perfect instrument. I think that there are bullet points to the right here that need to be also taken into account. And because this is a synchrotron talk, the bottom The bottom one is the one I want to focus about, which is the understanding of the measurement itself. So much work is done in this field using techniques such as mass spectrometers, which require empirical corrections, where you put a model aerosol inside and then try to, based on how it behaves, understand your complex mixture, which is the best we've been able to do up to now. But I feel that synchrotrons, and specifically some of the techniques that we will go into soon, can create a paradigm shift where we really understand exactly what's going on in the measurement and do no longer need to use proxies for quantification, because that really doesn't work on a complex mixture, because you don't know what you should compare it with. And all interesting aerosols, from my perspective, are complex mixtures. So when I was first contacted to talk, this was the title, Atmospheric Pollutants Interrogated by Photons. And I really like it because we use that a lot. And there'll be a few slides now explaining how to form a background before we get into the synchrotron results, which is going to be 5 minutes or so from now. So we have these techniques, the max synchrotron aerosol sample delivery system, which is the main part of the content here. And then we have an aerosol mass spectrometer, which is something that we use to characterize secondary organic aerosol particles and other particles in terms of their bulk composition, so the entire particle. And the point of the aerosol sample delivery system is that, as we will go into soon, it enables measurement of the outermost nanometer of the aerosol particle, which is very interesting to contrast with the bulk, because the outermost nanometer is the one that the surrounding gases will see. And to the extent that the particle doesn't dissolve, it's also the outermost nanometer that your body will see, your cells in your lungs when they deposit there. So they are of special interest. And as a background also, I will show some data which highlight various investigations we did and components of atmospheric aerosol that we soon will see how we can study them at MaxLab. So for this, we have also a multi-metal monitor, an XRF filter-based, which can be field deployed, and also a single particle soot photometer for laser-based investigation, black body radiation from individual particles, as well as a soot monitor, just light extinction monitor at allometer. Okay, so X-ray spectroscopy. You have basically 3 flavors of it here. And the cartoon to the right is illustrating the underlying principle. So you shoot electrons or shoot protons of well-defined energy from a synchrotron source, and then they interact with the atoms in your sample, in our case, aerosol particles, and processes take place, either absorption of photon, which may lead to just a depletion of that light wavelength, as you can observe, which we will see soon. Or if you do it in a way that it kicks out an electron, you get electron carrying information about surface layer. Or you can use X-ray fluorescence, which is good for elemental, establishing which elements are in the sample. Okay, the main one for this talk is the X-ray electron spectroscopy. And it uses the photoelectric effect that someone else got a Nobel Prize for, well, it was 100 years or even more now. So that's old news. And it works by having photons impact on the electron, and then the electron gets the energy and it gets thrown out. But if it is not sitting in a very surface layer of your sample, it's going to lose energy in some uncontrolled way, and it will not contribute to your signal. And for that reason, your signal is specific to the surface. And what you get from this is an information about the atom from whence the electron came, because there are little shifts there depending on the chemical bonds and other circumstances that this atom feels. And for that reason, we had, for a few years back, a postdoc pictured here, develop a sample a sample system, which we published last year. And it basically is an aerodynamic lens, which is the same technique that was used for the online mass spectrometry for some time now, 20 years or so, a couple of thousand publications on that. I think more than 1,000 records pertaining to secondary organic aerosol. But instead of a mass spectrometer, we have a photon beam from the synchrotron, and then an electro energy spectrometer, SIENTA, to record the kinetic energy of the electrons ejected. And why did we feel that it was a good thing to have this as a project? Well, because if you do aerosol science, you must do it on aerosol for practical purposes. I mean, some aerosols, especially secondary organic aerosols, have semi-volatile components which abundance in the particle phase will be dependent on the concentration and the surrounding gas. So it's an equilibrium that you ruin when you collect the aerosol. And on top of that, there is also the concern that if you shoot powerful X-rays on a sample, you'll ruin it. It's known as beam damage. So to get around these two problems, we created this, putting the lens and making a particle beam and overlapping it with the X-ray beam, as indicated in the cartoon. A key advantage is also the ability to study things in real time. I mean, you can study a dynamic process as it evolves, and it's also much easier to tune things because the experimentalists can get the feedback instantaneously. You don't need to wait. So you can correct your experiment on the fly, so to speak. As a background, here is another X-ray based technique and some pictures from a master thesis a couple of years back. I think this one would be supremely useful to study African air pollution, and I've tried but failed to fund projects which build on that. So it basically is a field deployable device that collects on a filter tape particles and then irradiates them in one step, and it produces elemental time series like these ones. These are actually from just my backyard in southern Sweden where I live. And you might wonder, why do I have a couple of micrograms of lead in plumes? And that is because this particular campaign was at a lead recycling plate. So it's quite useful to study point sources. Here, to move from metal to soot and organic aerosol and salt aerosol, which will be the focus of the rest of the talk, is an example of data from mass spectrometers in also my backyard, basically. So we have a rural station which is located about 70 kilometers away from the nearest urban center, Malmö, city of Malmö, 300,000 inhabitants. That counts as urban where I live. And we see the top panel here showing soot. So you see that the urban one is full of transient soot plumes. These are the cars. This is about a week or so of data, a little bit more. And then a bottom panel which shows the other components. And I want to highlight the organic and the sulfate. So the organic is the least understood one. Everything else is one specific salt or a couple of salts. And the organic one is maybe 10,000 different molecules with different origin and different effects. And the sulfate I want to highlight because it's often taken as a archetype for an inert aerosol. So an aerosol that does nothing, basically, as far as aerosol science is concerned when it comes to chemistry inside of particles. Yeah. And then to round off the background slides for the aerosol background, some results from experiments we did in our lab a couple of years back, where we measured, this is a 3-stone setup, stones I found lying around the lab, to be honest, but we also tried a number of stoves, and we connected it to the equipment we had at the time, which was not including the sample delivery system. So I would really like to do this again now that we are up and running with the synchrotron-based aerosol measurements. Here are more pictures from how it looks when we do this. So we have basically a volume where we have our combustion source. This, you can see my colleague Christina and her student Asmama here standing by the volume, and interacting with the combustion source, lighting a stove probably, and there me proposing by the mass spectrometer, which has been the main tool so far for me. Here is what it looks like inside the volume. So here we are testing an Ethiopian stove with charcoal, and we are collecting the particles for offline analysis, in this case mostly toxicology. This is a graph from the experiments, showing on top the gas, and then middle panel the light absorption, which is predominantly at short wavelengths and rather low in intensity compared to the mass, which is featured on the bottom panel. And here the line is supposed to represent total mass, and the shaded fields are supposed to represent subcategories. And as you can see, they do not agree, which brings me back to the first point of understanding and controlling a measurement. So this is an example of that not happening. The black line is in fact unphysical at points because it goes below zero. You can have negative mass of particles. Yeah, this is a reference instrument for measuring PM. It's an oscillating filter, and it has problems with volatiles. That's why it gets negative there. Some of the background for my motivation why I like to do my aerosol science with synchrotrons moving forward. Here is a picture of another type of stove, a Swedish wood stove, which is, from terms of pollution, a very potent maker of polyaromatic hydrocarbons. We saw very little of that, comparatively speaking, from the African stoves. And this has to do with the creation of underventilation and very hot combustion inside of this. You can think of it as a heating wood stove. And it connects to the amount of oxygen in the flue gas there. And what we did also with the wood stove is we use a reactor to simulate photochemistry. So it's basically just UV lamps that shine light on the aerosol as it's emitted. And then we observe two things. We observe in the green squares on the left axis what's known as enhancement. That just means we get more. So if the enhancement is 4, that means that when we aged it, it became 4 times more abundant, which sounds strange, right? But we're talking specifically about the particle phase. So that means that more gases have been oxidized and started to contribute to particles. And then as we go further up to a higher setting of our PAM reactor, which creates this secondary aerosol, we see a decrease in the amount of secondary organic aerosol because it starts to fragment the molecules and drive them towards species that are more volatile. Consistently during this particular experiment with the Swedish stove, we can see that the oxidation state of the carbon is going up. Every time we add more photochemistry, we get a higher oxidation state, which intuitively makes sense as a background for later things. So here is the first stuff we did with the aerosol sample delivery system, which pertains to secondary organic aerosol. So here we use a reactor called the organic coating unit, which you can read about there if you're interested, to oxidize alpha-pinene, which is a gas and a known precursor. And on the top panel of the graph, we see the same oxidative state of the bulk, again, going up consistently with more lamps. And then we see in the middle panel, the top panel is the surface. Yeah, and it goes up consistently like in the Swedish experiment. So that was in line with expectations. But when we look at the middle panel and study the bulk composition, it actually doesn't oxidize for the higher steps. It gets less oxidized. So there's a discrepancy here. And I want to think that it's related to the very high amounts of secondary organic aerosols that we are producing at that stage, because this influences the partitioning of the formed species. Again, this is the setup, and you see you can put any aerosol in the green where the green arrow is, and then the X-rays are going to hit it, and then this round part up here is where we get the electron energy measurement via aerodynamic focusing. This particular slide is a repeat from a meeting we had in Addis in 23, just after this first pilot experiment. And it shows my view from the balcony at the time and the effect on this oxidation. Now we're seeing the spectra, how they look like when we do this. And you see that you get, this is not what would be known as a perfect instrument with molecular information. No, it is a way of classifying different functional groups that we have, which, to be honest, is a bit of a relief because I don't have chemistry training. I'm trained as a physicist, so I can't deal with all those molecules anyway. Here is a more extensive data set that we got the year after. Here we have tried to do the same thing, but using a reactor which employs a bit more slow oxidation. I would call it less user-friendly, more atmospherically relevant. This one is known as the GoPAM. It is published as well. And you see the electron spectra on the left-hand side here, divided by precursor molecule, left or right. So we have alpha-pinene as a representative of biogenic gases, and then toluene, which is emitted in combustion. So you get it from stoves, you get it from cars, etc. And then we have varied number. Here we keep the oxidations constant. So the sun would be just the same sun all the time. But we do change the amount of precursor, which does influence the amount of particulates formed, which does have some role on the spectra of which relate to us information about the surface composition. But the interesting thing here is what was more impactful than the amount of precursor or even the type of precursor is, are these particles wet or dry? Because this type of experiment is usually done by providing a seed aerosol. Sulfate is the standard choice, and then having things condense on that seed. And I had a notion that the stuff that's happening in that process on the surface of the seed might be important for the composition of the SOA that grows, and this plot confirms it. When we compared it with the pilot data, which is down here in this summary plot, we find together they give an inverse relation where the kinetic energy, it goes up with the O2C. So kinetic energy of surface electrons goes up with the bulk O2C ratio, which is confusing because higher kinetic energy means lower oxidative state. So these two are going backwards, and work is ongoing, trying to figure out why and how. Here is another example done by my colleague. This slide I actually borrowed from Antti for the talk. I showed the balcony picture from before because he had just given a talk at the African Light Source Conference in 2023. And I was very happy to report that our work was mentioned there. So when Antti made the slide, it had just happened that my colleague had run the first beam time, the first 1 week-long experiment looking at soot. And that was a first for MaxLab because no one before had had an open flame as their study object. So there was some safety forms to fill before we were allowed to do this. And it opened the way to do other things. Solid fuels is, I think, from Africans' perspective, the most interesting, or just from aerosol science perspective, the most interesting combustion source. So I can envision projects where we do experiments like we did with the mass spec, but in this environment instead. Okay, so what my colleague Con has done afterwards, it's of course to analyze the data, and there's a little taste of it in a recent PhD thesis, which I have a link for here, but that's just a taste because the papers here are coming in the coming year. We usually do a little teaser for coming work in theses in Sweden. So what was done was the sample environment was used to study particles extracted from a flame. And we do this via a probe, which is indicated here to the left, and it just sucks air out, and then We get those particles, we move them to the sample delivery system, and here we have varied the, we can either vary the flame height, the height in the flame, which is the maturity. It starts low and it matures as it goes up. Or you can vary other things, for example, the mixing ratio, so the leanness of the flame, which has been done here. So this one has a fire of 1.77. It's a fat flame, lots of fuel, not so much oxygen. And then as we reduce this ratio, we see more oxygen containing functional groups appear up to a stage, and then soot maturation processes proceed. So we get different spectra for different sampling types, and work is ongoing to make a story out of this. Another example of aerosol science done with the sample delivery system is this recent paper from earlier this year, where we are looking at brines from Tibetan lakes. So our colleague Kong, he had sampled from there and sprayed a lot of bottles of these different places, the different brines that were collected. And then what Kong did was a trick called depth profiling. So if you change the photon energy of the incoming photon, you also change the kinetic energy of the escaping electron. And the escaping electron energy has implications for the attenuation in the material. So by doing that, you can selectively look, I mean, instead of the first nanometer, the third, or the, you understand the concept. So this is referred to here as penetration depth. And we see that the different components of the salt, they layer differently. So these particles, just like the secondary organic aerosol particles, they have internal structure, which we can quantify with this technique. The take home from that study was that even though the amount of sulfate was higher in brine number 1, if you look at the liquid and the sulfate content in the liquid overall, when we make particles, we actually get less surface sulfate, which is interesting because the surface is what participates in the chemistry, what's going on. So if you assume that your surface is the same as your bulk, you will be severely misled. Here is another example of using the sample system to study salts. So we are mixing here sodium chloride and ammonium sulfate because according to basic inorganic chemistry, this should lead to a recombination of ions where the sulfate it's an ion pair switching, which releases the chloride from the sodium chloride in the form of hydrochloric acid. So it actually turns out that the sulfate under these circumstances, which are not unusual at all, these are amongst the main atmospheric components, ammonium sulfate and from SOx oxidation, and then sodium chloride from sea salt. It actually turns out that when you dry these things, you drive out. So there's chemistry happening between these supposedly inert components. This is a cartoon illustrating basically what's going on in the picture here, that when you dry it, depending on if you saturate the gas phase or not, you will end up with different particles in the end. So ammonium, the sulfate will be in different forms. Here is a final, how am I doing? I think I have a minute left.
You have a minute left, yeah?
1 minute, okay. A final example of something that's been done is spraying of engineered carbon nanomaterials by my colleague Wilhelm. And he did this because he's interested in the toxicological effects on those surfaces on humans, which we try to proxy using this instrument, which measures oxidative potential real time. So here it is combining this toxicity probe with the surface characterization in one measurement, which is live. And then we can ask ourselves, is the proxy good enough for the health? But that's a later question. This is just a point that we can take aerosol and sample them live. We can also take them and get them back in the air and study them that way, which opens up to importing aerosols, right? I think in the interest of time, I'm going to have to skip the absorption spectroscopy, but believe me, it's also useful for aerosol science. Here are my take-home messages. I hope there's time for a question.
I'm just reading through the take-home messages. Are there any questions for the speaker? I think we should have a school on air pollution, aerosols, and synchrotrons.
I would like that.
There's a speaker.
Can it be next year?
It can be, especially if we do it remotely.
Yeah.
What my feeling, it takes 2 years to make something happen in person.
I'm under some pressure to organize something like this next year.
We can make that happen. I guess you can do it in person in 1 year, but you have to have a ton of money.
Yeah. Well, that's the thing. We need to spend the money next year, you see. No, it's not that kind of money which enables. But I mean,
got that kind of money.
Hybrid thing could be an option, perhaps.
Yeah, we can do that. So let me ask you just about in aerosol, there's the particle size.
Yes.
And particle density. And what are your upper limits in those two things? And I guess they are somewhat related. I guess they are related.
Oh, do you mean particle density as in material density of the stuff?
Right. So the aerosols, there's a solid particle entrained in some gas. So how many particles per unit volume?
Okay. So you mean that in that sense. Okay.
Yeah.
That is actually a limitation in the other end, that we need a significant amount of them to produce spectra in reasonable timeframes. That is the focus of our development work on this system now, because there are several bottlenecks identified having to do with the collection of the electrons, for example, and also the O-beam overlap and other things. So that is not a problem that there's many. Well, it becomes a problem when we start clogging things up. But generally speaking, we are more concerned about the lower limit. When it comes to size, this is a serious limitation that we're also working on overcoming, because we are just using a commercially available aerodynamic lens, which is optimized for sub-micrometer particles, but not ultrafine particles. So they need to be, if they're over 100 nanometers, they're plenty big enough. If they're 50, it's difficult. That's where it starts to hurt. And the upper end, we are also, when we go up towards the macrometer, a little bit limited due to the specific lens that is installed. Other lenses are available, so it is a matter of installing them instead. Yeah.
Okay. Have you had any projects that looked at dust from the Sahara Desert that lead to the African easterly waves, which lead to hurricanes in the Western Hemisphere?
I would like to have such projects. I started thinking about them, but I haven't even written the proposal to be rejected yet. But the dust is supremely interesting because it is what brings most surface into the air. I mean, as a system which is specialized on surfaces, it certainly should look for dust applications. There's much happening with the chemistry on dust surfaces.
Right.
Of other things. So yeah, that would be that's something I'm actively looking into.
Okay. Let me check and see if there's any other questions for you before we move on. Maybe not. But yeah, let's keep in touch and so we can do like a Zoom school. That seems infinitely doable. And then
I'm going to hold you to this.
Yeah, and then when we get, unless we find a rich uncle somewhere, we can in 2026 we can do it. We can contemplate an in-person school, maybe in 27 or 28.
This is a wonderful suggestion. Let's talk offline.
Okay. All right. Well, thank you for your time, and let's move on to the last speaker, your colleague, Jorgen Larsen. And by the way, you mentioned Malmo. I've actually been there a couple of times.
Oh,
yeah.
not many have.
Yeah. Okay. Thank you for your time.
Thank you.
So, Jorgen, how are you today? Am I pronouncing that correctly?
Well, I think you pronounce it very well.
Thank you.
So, yeah.
Okay,
So,
let me
take your
allow you to share your screen. Hold on one second.
see if I can get that going.
Yep.
So did that come out all right?
Yes, indeed.
And then I'm gonna have to find this anyway. Thanks
Okay.
for allowing me to present the Femtomax beamline. So just like my colleague, Axel, I'm also a professor at Lund University rather than employed at Max 4. I have a few different duties at Lund University. So I'm co-directing an institute known as Links, and this Links is a place where we try to facilitate the use of both Max 4 and also the European spallation source, which is being constructed right now here in Lund. I don't know if you actually can see my pointer as I'm going ahead on the screen, or I need to do something here.
I can see it. I see it. At least I can.
Okay, great. Then I'll continue. And but today I'm talking not as a professor or as a co-director of Links, but I'm talking as a member of the Femtomax beamline team. Femtomax is not on the storage ring of Max 4. It's a Linac-driven source. There is a full energy injector. The finest beam that we just heard a talk about is located in this smaller building here. And that's also injected from the same Linac. But when they're not filling the small ring here or the big ring here, they send very short pulses to us over at the underground at the Femtomax beamline. So what we're doing over there is we are looking at ultra-fast structural changes. So the closest thing we heard so far during this meeting, I think, is the talk by Petra Fromme, who is giving a more biological studying more biological system, whereas we are studying more physics related problems like phonons, phase transition, perhaps shear wave propagation. And the unique thing about our beamline is that we can study dynamics on 100 femtosecond time scale. So pump and probe experiment goes like this. Electrons are accelerated through the Linac, and we need to synchronize a laser 300 meters away. And when we do that, we can probe one time frame of what is known as a molecular movie, where we can see how a system evolves with time following laser excitation. So looking at it from above, it's a pretty large facility. It's 300 meters. It's not like the AfLS, which are kilometers. We can still do hard X-rays, but at a lower flux, but also at a significantly lower cost, because these linear accelerators are for injecting the rings. And that means that the additional cost is building the beam lines that go with it. We need also to have a photogun, which is an investment that would not be necessary just for the rings. And we need to compress the electron pulses to 100 femtosecond, which would not also not be an issue. So if you go to an FEL, you will find 100 meters or so of undulator undulators. At Femtomax, we have two sections of the SACLA undulators, and they're 5 meters each. So we have 10 meters of undulator. It's not enough to get lasing, but we can still get relatively high flux, 10 to the 7, 10 to the 8 photons per pulse, coming in a range between 2 and 15 kilo electron volts. Otherwise, the beamline is pretty much standard. We can focus the beam. We can choose different monochromators to have different bandwidths. The other different thing compared to the ring beamlines is that we don't have 100 megahertz, so we have 10 hertz. So the average flux is comparatively low, but on the other hand, we get the very high time resolution. In order to initiate reactions in solids, we have a laser which is situated on the ground level, whereas the beamline is radiation safe way down in the basement. So this is what it's about. It's a cartoon of what it looks like. Our X-ray pulses arrive 10 hertz. They have to be synchronized very well within a few hundred femtoseconds ideally, but in reality, it's maybe about a picosecond or so. So the pump and probe, the pump initiates some reaction in the solid. We probe it with hard X-rays, and we can vary the time between pump and probe for different shots and see what is going on inside the material. And the ultimate time resolution is limited by the duration of the pump and the pulse probe. And you can see the reference to my former PhD student Henrik Enquist, who was responsible for much of the synchronization work for the Femtomax beamline. There are a few classes of experiments that we have opened up for user operation. We do in-air diffraction. We sort of just do different types of scattering at the moment. We are using a cryojet to cool here for a sample that needs to be cooled, and typically we use this cap goniometer and place a Pilatus detector on a robotic arm in order to measure the dynamics. We also do in-vacuum diffraction in this vacuum chamber. The sample position is around here. There's various types of diagnostics that we can place inside the chamber. The detector is mounted in a fixed position here at the exit of the chamber. And it allows us to study a range in Q in momentum transfer between 0 and 5, roughly, inverse angstroms. We can manipulate the position by placing an attocube stage here, and we can cool. We've been doing experiments all the way down to about 10 Kelvin with liquid helium, and we can heat the sample to 500 Kelvin. We have different types of detectors. We mostly actually use the Pilatus for almost everything, even though the pixel size is significantly larger than on some of the other smaller detectors. We have different pump sources. We can use the Sapphire laser itself. It provides about 6 millijoules on the sample, 50 femtosecond long pulses. And we can change to an OPA, which can provide visible radiation or deeper infrared radiation. And this one is sometimes needed in order to match band gaps or other triggering mechanisms. We also can interact directly with with molecules without heating or without exciting carriers, and by generating terahertz radiation in organic crystals, and then transport focus the terahertz radiation onto the sample and probe that area with X-rays. So these are the pump sources that we so far have available for users. Now, I mentioned before that we like to have 100 femtosecond time resolution, but in fact, the synchronization between the lasers, which are 300 meters apart, so that gives the synchronization between the X-ray pulses in the laser, it's just a picosecond. So it's not good enough for us. So for that, we are measuring the relative arrival time of the laser and the X-rays for each individual pulse at 10 Hertz. And exactly how this is done, I don't need to go into that maybe, but I can show you that if we don't do this post-sorting, we have carried out an experiment where we looked at optical phonons, in particular the AG phonons, which is excited by the laser and then probed by bismuth. And it's very common, like fruit fly experiment for ultrafast science. When we don't do the post-sorting, we see something relatively dull like this. But when doing the post-sorting, more structure emerges as our time resolution goes down towards 200 to 300 femtoseconds. Now, that isn't good enough either. So we have another timing tool, which is a cross-correlator. That means that we extract visible light from a bending magnet and then do a so-called cross-correlation with the laser in a nonlinear crystal. That allows us to get a signal which really has a high resolution. And I'll see if I can get my little movie going. So you can see that this jumps about, and each position is half a picosecond, like each line in the diagram above. And we can pin this down with an accuracy of about 50 femtoseconds or better. So that means that we can eliminate the jitter by measuring each pulse using this device known as the cross correlator. And it actually works. This is the fastest dynamics we've seen so far. This is something I'll talk about a little bit later as an example of an experiment we carried out. And it's actually material called GST, where we look at diffuse features. And it's actually the same signal with either the first timing monitor I showed, which could give such a nice results for bismuth. And then below, we are using this new cross-correlator tool that was very recently commissioned. And you can see the time resolution is improved by at least a factor 4 or so, down to 70 femtoseconds, which is the best we've seen so far. We came about to do this when we were really looking into maybe being able to investigate what could be a very fast phase change memory. So if you're in my age, you remember using DVD records to write and read information. And inside your DVD writer, there was a laser. It would be heating up this material a little bit. And if it heated it up slowly and then let it melt, it would grow back to a crystal. But if you did that more rapidly, and now we're talking not femtoseconds, but maybe milliseconds, then you would get the amorphous phase. But that is not really what we're aiming for here, but we would like to be able to read and write in less than a picosecond. And we're not there yet, but we're investigating the processes using FemtoMax. And the idea is to use something which is called non-thermal amorphization. So what we mean by non-thermal is that we don't really rely on melting. We are inducing a process perhaps in the material which brings it to the amorphization as more of a coherent motion. And that can happen when you break bonds in material. So we break bonds with the laser, and then we would like to see it evolve to the amorphous state. So we are particularly investigating this material, DST, that was used in the old DVD records. But I'll jump past this one and just say that we're not the first who have been trying to investigate this. So, but we are getting better and better tools, and FemtoMax is a very nice tool for this type of experiments. So we are doing powder diffraction. These are powder films that you have on these samples. To the left, you see the setup. with the laser in red, the X-rays in blue, and you also see what the structure looks like on the amorphous phase, because this is what you get when you just grow the films. These particular films are 30 nanometers thick. If you heat them up to about 130 Celsius, then you get into the crystalline shape, which we have to the right. And then we would like to study the transformation to the amorphous phase as we illuminate it with the laser. So being able to write 0 if we have a 1. There's been one particular model which really spawned all these experimental studies, and that was by Kolobov and co-workers who proposed the non-thermal amorphization model called the umbrella flip. So it's a germanium atom that moves from its regular position to start a more tetrahedral motif, which then transfers to it looks like more like the amorphous phase. Now, what we don't believe that we will ever be able to make a coherent motion that does everything at the same time in the same direction. But there are many germanium atoms, and they can move in different equivalent directions, and possibly also at slightly different times. And this is what our preliminary results are. We're in the process of writing this up, but I thought it was a nice thing to show that if we modify the umbrella flip model slightly, so that when the germanium atoms in the unit cell, see if I can get my cursor going, moves into the cell like this from this position in here to roughly a quarter, then the tellurium atoms are not bonded to this anymore, and they would move slightly. And if we introduce these models, we can actually simulate. These are actually simulations, the solid lines, using this relatively simple model, and they fit to the experimental data fairly well for 5 different reflections. So I see I'm not doing super well on time, but again, I can show you that this is part of what we are looking at with the cross correlator. We can really see very fast dynamics. You see some of it also here. These we believe are actually breaking of the bonds and rearranging valence electrons that we see as a trigger mechanism before the motion occurs. Now, we really want to see the amorphous phase. So this is what it looks like before we have annealed it to the crystalline. So what we do then is we fire a laser onto this one, and when we have fired the laser, it doesn't really look much different just by eye, but we can subtract the 2 phases and see if there is anything remaining. And this was our very first attempt at femtomax that you see here. We didn't think it was good enough data, so we tried to improve the Q resolution by reducing the beam from about 18 micron at the time to 20 microns. And what we see here is that the blue lines indicate that the crystalline peaks have been reduced, whereas the yellow here indicate that there is something growing in between. So we had some hope for a while. What we then did is that we take one of these images that would be one particular time point. We make call it like you sum all of these different contributions, all these pixels, and we turn them into one point. So but then on the other hand, we take many of these pictures at different times. And then from data looking something like this, with enough shots, we indeed get something more like this, where we can see again that the crystalline sharp peaks disappear and something else is growing. Now, this is what it looks like. We've just do a line out. This is what the structure factor looks like before excitation and 1 picosecond after excitation. Comparing these, subtracting these would then give the what has changed, and this is then what could be the what we first hoped would be the amorphous phase, but we are comparing it to the data that has been recorded before, and it doesn't fit very well, as you can see. There is, of course, a stronger peak here and another peak here, but they don't seem to be in the right place. However, in this very short time of the picosecond, the density has not time to change. So it's something that we are currently working on to try to understand how does the expansion of the lattice occur on these very, very short time scales. And is this actually the amorphous phase at the density of the solid material? So to summarize this part of the talk, we've seen the ultrafast reduction of the crystalline shape. We have a modified umbrella flip model that can explain the intensity of a number of peaks. We have seen the ultrafast appearance. This is what happened in 70 picosecond that I showed you earlier on for the cross correlator. So that happens very quickly, but we have no clear evidence yet of amorphization, because essentially the pattern doesn't fit with the amorphous phase. There is a bunch of collaborator here from both from physics, from near-lying universities, from Max4, and collaborators in Rome who made the samples for us. I just want to say while I flip through this that we are now open, Femtomax is open for general users. There's been a lot of interesting work done, and just flipping through. And we hope to be able to see perhaps even users coming from African universities in the future, and stronger collaboration. So thank you for your attention.
Thank you. It's interesting talk. Interesting. Let me ask to see if there's any other questions before I ask my 1 or 2 questions. For the what did you call it? Umbrella flipping, umbrella inversion. So that's not a 2-color experiment where you're looking at IR. You don't have an IR.
So the IR would be inducing the umbrella flip, whereas we are probing it with the X-rays. And of course, in the end, we're not really interested in probing it. We want to make sure that we can make a flip every time and then write the information in a picosecond, which would make a really fast storage medium.
I see. I was sort of interested in the other way around, using the X-ray to cause the flip and then looking to see what IR bands appear or disappear.
So there are actually, I mean, there are a lot of work going on right now with building X-ray pump and probe end stations or at various places. But it's a bit tough for us. We have a relatively low X-ray flux compared at least to the free electron lasers.
Yeah.
But again, it's something that we could afford at Max4 at this time.
Now, this strictly speaking, that that's not lithography. That is just what? Because you're not leaving an etch in the substrate, are you? When you
No,
do
I
the
mean
umbrella?
it's so you can think about it as when you were using the good old DVD records in the old days. These structural changes between the two phases were occurring then as well, just by heating, melting, and then regrowing, which then took him something like a millisecond or at least hundreds of microseconds to achieve. But you didn't have any lithography at that point either. You could just write in some place on the record.
All right. Okay. Now, so Does your beamline have any schools or workshops contemplated in 2026?
There is likely to be some workshop in connection with our users meeting, but that's already in January.
Yeah.
Schools, we are not planning. We often take part in the Hercules school,
right,
which was mentioned before from ESRF. There will be actually 1 week. It will be hosted by Lynx this year.
Okay.
But here in Lund, whereas the other time is in Grenoble.
Got it. Yeah. Oh, you're right. Right. Yeah, that's right. With Hercules. So with Hercules, that works out. So I guess when the students fan out to Hercules, it's
exactly.
There has to be a beamline that says, "Okay, we'll handle this for a week or whatever, however long."
Right. So people actually then further fan out when they get to max 4 to the different beamlines. So we typically have like a small group of 5-10 students or so that we train on these techniques. We also take project workers and exchange students and so on. Somebody wants to write master thesis and so forth.
Okay. Well, I'll try to round up some students for you. January might be a little too quick for your users meeting, but we'll figure something out.
But there's also January 2027.
Yeah, yeah, and I'll be here, so
yeah,
for sure. Okay, well, thank you for your time today. And
thanks for inviting me.
If there's no other questions for the speaker, then we will close the session for today. Yeah, I think everyone's sort of fried out, I guess, so it's pretty late. Okay, well, thank you very much for your time. Okay, talk to you later.